University. Yun. Philippines Science High School, Caraga. Northeastern Mindanao State University. Surigao del Norte State University. Villa Cananga Integrated School. Department of Environment and Natural Resources, CDD Caraga Office. So the following are the government agencies. Okay, thank you for being with us. And also, likewise, to City Environment and Natural Resources, City Government of Butuan. Are they here? National Museum of the Philippines, Butuan. Okay. And of course, the Philippine Statistic Authority, Butuan. Thank you. So before we begin, I would like to remind everyone that please keep your phones on a silent or vibrate mode. So you are also welcome to take photos later and share your favorite moments from today on social media. Friends, the Philippine Biodiversity Data Congress 2026 is now about to begin. As we open this Congress, let us ground ourselves in a spirit, gratitude, and purpose. We invite everyone to rise for the opening prayer, followed by the singing of our national anthem.
Amen. O God, by your grace, your servant Father Orius, enkindled with the fire of your love, became a burning and shining light in your church. Grant that we also may be aflame with a spirit of love and discipline. May we ever walk for you as children of light, that our lives may reflect your loving radiance, and let your light shine. In every word that we say, in every action that we do, in every choice that we make, and in every noble cause that we commit ourselves to. Through Jesus Christ our Lord, who lives and reigns with you in the unity of the Holy Spirit, one God, now and forever. Amen. In the name of the Father, and of the Son, and of the Holy Spirit. Amen.
Ang bituin at araw niya kailan pamay di magtitingin.
be seated. And now, ladies and gentlemen, to welcome us to this Congress and to provide the rationale, we are honored to call upon our Vice President for Academic Affairs and Research at FSUU, Dr. Arlene M. Floreta.
Good morning, everyone. Those who are joining us physically here on Father Saturnino Orius University, CBE Function Hall, maayong adlaw, good morning sa tanan. And for those who are joining us live via FSUU Butuan Facebook page, greetings everyone, maayong adlaw from FSUU. I am so honored and grateful to welcome everyone in this Philippine Biodiversity Data Congress 2026. This Congress is an initiative of the Biodiversity Informatics and Research Center of Father Saturnino Orius University. This was established in 2016 with a mission of documenting rich biodiversity of the Caraga region and generating research that will address both local and national environmental challenges. Today's gathering is anchored on the theme Field to Cloud, the future of open access conservation. Our country indeed is recognized as one of the world's biodiversity hotspots. Yet, despite this richness, conservation efforts are often constrained by limited access to reliable data. At times, the failure to protect certain species is not really rooted on neglect, but due to lack of awareness of their ecological significance. So today's Congress seeks to bridge that gap. The program highlights areas such as real-time data collection for biogeographic and evolutionary research, 3D imaging in functional morphology, and impact of technology on the conservation of iconic flagship species, and field-based long-term conservation. Central to this is the field to cloud approach, which promotes open access systems, ensuring that locally generated data are not only scientifically robust, but also transparent, sustainable, and accessible to the broader conservation community. In doing so, we support more informed and evidence based decision and policy making. In parallel with our plenary sessions this morning, we have curated engaging and experiential learning opportunities. At the lobby right now, participants can take part in a simulation of field experiential learning and biodiversity documentation, where the participants will engage in field data collection, proper documentation, analysis, and ethical data management. This activity is facilitated by Ms. Daphne Kyle Mbahoy, our biologist working under the Mabolig project of the Department of Science and Technology. On the second floor of this building, there's also a workshop on digital imaging, photogrammetry, and 3D modeling, which is conducted by Mr. Dominic Argiritan, the project lead of the Mabolig project under the OST. This workshop demonstrates how advanced technologies can capture and reproduce biological specimen with remarkable accuracy, reinforcing the integration of innovation in conservation work. We are also privileged to have distinguished lineup of plenary speakers who will bring global expertise and insight. We have Dr. Rafa M. Brown, an evolutionary biologist and herpetologist from the University of Kansas, whose work on species diversification in Southeast Asia has led to the discovery of nearly 100 new amphibian and reptile species. We also have Dr. Duncan J. Ershik of the University of Massachusetts, a biologist and innovator known for translating biological principles into technological applications, including bio-inspired materials and 3D modeling systems. We also have Dr. Mariano Roy M. Duya of the University of the Philippines, Diliman, an expert in mammalogy and vertebrate ecology, whose research contributes significantly to the understanding of Philippine biodiversity. Dr. Jason S. Ibanez of the Philippine Eagle Foundation, a leading figure in eagle ecology and conservation, known for advancing habitat protection through science and community-based approaches. And finally, we have Miss Lisa J. Paguntalan, the Executive Director of the Philippine Biodiversity Conservation Foundation Incorporated, an accomplished ornithologist whose work has led to remarkable species rediscoveries and strengthened community-based conservation efforts. Indeed, today's program is very rich with opportunities for learning and engagement, from insightful plenary discussion to hands-on experiences in emerging conservation technologies such as photogrammetry and 3D modeling. This only reflects the innovative directions we must take to safeguard biodiversity in an increasingly data driven world. We extend our heartfelt gratitude to Dr. Marites B. Sanguila, the coordinator of the Biodiversity Informatics Research Center of FSUU, whose leadership and vision have been instrumental in establishing the center as a vital force in biodiversity research and conservation in our region and in the national level. To our students and faculty, researchers, government partners, and other stakeholders, may this Congress serve as a platform for collaboration and strengthen commitment to ecological stewardship. Once again, warmest welcome to one and all to Philippine Biodiversity Data Congress 2026. Good morning.
Thank you very much, Dr. Florida, for those thoughtful and inspiring words. Your welcome message reminds us all why we're here, not merely to exchange data, but to share a collective commitment to the natural heritage that defines our nature. Your vision for research driven academic excellence has set the tone for everything we will do today. We are truly grateful. And now, moving on for the intellectual heart of this morning program, in an era defined by rapid environmental change, the ability to document, digitize, and democratize the biodiversity data has never been more urgent. The scholars we have with us today are at the very forefront of this frontier, pushing the boundaries of science and technology, precision, and passion. Our first speaker joined us today from the United States of America, and yet his work has brought him closer to Philippines shores. He is a professor and curator at the Biodiversity Institute of the University of Kansas, one of the world's most prestigious natural history and biodiversity research institutions. For decades, he has conducted intensive fieldwork in the Philippines, becoming one of the foremost authorities on Philippine herpetofauna and island biogeography. His research integrates field survey, museum collections, and genomic data to illuminate evolutionary history and geographic distribution of life in our archipelago. A contribution that has not only enriched global science, but has directly informed biodiversity conversation efforts here at home. Today, we will, he will speak us today about real-time data collection for biogeographic and evolutionary research, a topic as timely as the transformative. Ladies and gentlemen, please join me in welcoming our first plenary speaker via Zoom, professor and curator of Biodiversity Institute of the University of Kansas, Dr. Rafe M. Brown.
Hello, everyone. Hello, everyone. Okay, we have an echo. I hope you can all hear me okay. It is so great to be here. Thank you so much for the invitation. See if I can mute my speaker. Can you all hear me okay? We're going to just project. Please send me a chat message. This isn't going to. Okay. Hold on just a second. All right, let's try this. Nope, it's still. Hmm. Okay, folks. I can hear loud echo. Your end, can you? It's really kind of disruptive to me, but I'll ignore it if everyone there can hear what I'm saying clearly. Somebody send me a text and let me know if it's coming through at your end. In any case, it's so great to be here. You all, thank you so much for the invitation to share some of our recent results. Today, I'm going to talk about biocollections and the associated data from the field to the cloud. I hope everyone can hear me okay. I'm going to talk about the importance of use of biocollections and how collections are used in science. I'll discuss a bit the extended specimen concept and the way in which we share biodiversity data through distributed databases. I'll talk about scientific transparency, inclusion, and reproducibility in biodiversity science. And then I'll wrap things up with a discussion or relating how it relates to integrity and repeatability of Caraga region science and biodiversity conservation policy in the Caraga region. Checking the chat real quick to see if you can see the presentation. Okay, if everyone is clear on my presentation, we'll just go from here. I hear an echo, but if it's okay with you all there, someone give me a thumbs up by text if you can. I'll keep going. Why do I care about all these topics that I'm excited to talk to you about today? Points 2 through 5. Well, because I come from a really boring place compared to the Philippines. I hope that, okay, no more echo. Whatever you all did there, the technical, it's working better. Can everyone hear me okay? Let's see, a wave of hands from the crowd. Can you all hear me? There we go. Okay, very good. Text me if this isn't coming through. Compared to where you all are right now, I come from a really boring place. I'm zooming in today from the University of Kansas. This is in the middle of the central part of the United States. This boring state is a perfect rectangle of almost homogeneous habitat all the way from the east, all the way to the west. There's a little bit of an environmental gradient, but generally it's a continuous, homogeneous landscape that is decidedly boring from a biodiversity prospect, except that it's extreme opposite of where you are on the other side of the world in an island archipelago, where it all gets exciting and there's so much more to think about. For me, growing up in an area, this part in the central part of the United States, far away from the ocean, the idea of an island archipelago and the simple concept of how difficult it is for organisms to colonize and occupy faraway land masses would just seem like such an exciting and interesting topic compared to this view of the world of a flat, perfectly homogeneous landscape, where of course organisms can just populations can spread and where there are no isolation, there are no barriers to colonization of populations or spread of populations. It's the exact opposite in a way of the kind of archipelago environment that you're in now. We appreciate island archipelagos because of their simplicity. Many islands are very simple and only contain a few species, and that allows us to look at their natural history in exquisite detail. We appreciate island archipelagos in particular because of the replication factor, right? These islands are replicated units, and islands occur in in archipelagos where it's almost like a perfect little experiment in the sense that the size of the island and the distance from the mainland can occupy which species can occur there and how they've colonized and how they make a living in that part of the world. So it's almost an experimental biologist's dream to study biodiversity in an island archipelago where landscapes and environments are replicated. And of course, coming from this sort of very boring Midwestern part of the central part of the United States, I had really good mentors early on. So my journey involves a bunch of really good mentors who encouraged me to go to the other side of the world and follow my passion in studying biodiversity in island archipelagos. And so I want to talk about sort of what some things that I've learned in the last 3 years that I consider to be things that the Philippines taught me. So today I'll start initially by talking about the importance and the use of biocollections. And we'll start with the story of Edward Taylor, who exemplifies the Philippine-Kansas connection. Edward Taylor was a professor here at the University of Kansas, and when he was still a student, he traveled to the Philippines after he got his undergraduate degree, and he traveled to the Philippines and lived in Butuan, just south of where you are now. For more than a year, he established a boys' school and introduced American-style education at that time on the tail end of the Moro Wars in central Mindanao. He established a boys' school that taught American-style education, and he collected a lot of specimens. And so that interest in that specimen-based research really sort of starts with the attention to the Philippines in the United States, starts with this tradition of Edward Taylor, another KU professor from 100 years ago. That's interesting because it turns out he wasn't just a biologist, he was also a spy. And he was there spying on the Japanese encroachment into the Philippines. And he sort of had a double life, and there's lots of rumors and lots of stories about the drama that his life may have been. But during the time, one of his covers when he was being a spy was not pretending, but playing out the role of being a field biologist. And so he sort of his cover was being a herpetologist, and he collected and deposited specimens at the Bureau of Sciences in Manila that he brought back from Bunawan by schooner. And then, unfortunately, during the Japanese firebombing of Manila during World War II, many of those specimens were lost, and somewhere around 30 to 40 species are considered our lost Philippine species of the Philippines. They're species that the original specimens were gone. There was a literature publication about them, but the scientific community didn't know any more about them than the fact that there was these publications because the specimens were gone and no one had seen them since. So here's a view of the Bureau of Science in the early before World War II, which was a happening place with lots of activity and science right here in central Pearl of the Orient era of the Philippines. And here's a view of the Bureau of Science after the Japanese firebombing at World War II and an American Jeep parked out front. You can see that the entire interior of the building has been gutted and burned. And in addition to the Bureau of Science, we also, at that time, the Manila experienced this entire firebombing of Manila that resulted in the destruction of the old Congress building. I hope a lot of people here have traveled to Manila and have seen that building today, which has been rebuilt and has been the National Museum of the Philippines for the last 30 years. Here's a view of the National Museum of the Philippines, which used to house biology and art and archaeology. And what you can see happening here, this is a view from the Bayleaf Hotel across Rizal Park. You can see, across from Intramuros, you can see what's happening
Recording
here.
in progress.
You can see what's happening is a flood is encroaching into the National Museum. That represents a real threat to those collections that are housed there. And those collections in 2015, here's a picture that I took in 2015 of the collections there in the National Museum of the Philippines. Those collections were in the basement. So all that had to happen was this water escape into the National Museum of the Philippines, go down the stairs into the basement, and those collections would have been completely flooded, and that record of Philippine biodiversity would be lost. Fortunately, that didn't happen. Unfortunately, your former president, the late Pinoy, during an executive order, mandated the establishment of the Philippine Museum of Natural History. And now, to this day, you have a world-class museum celebrating Philippine biodiversity in Manila. I hope everybody here gets a chance to spend time at that museum and travel around and look at the amazing exhibits and the wonderful space that is created from that rebuilt architecture, of course, all of which was raised during World War II, and now that houses those specimens from back then. Those specimens form the basis of a research program of a number of curators and researchers there, but they're also on display for the general public to appreciate. When you go there now, you see these beautiful exhibits of Philippine biodiversity. It's a great thing. We've been waiting for this for 30 years, and I can't tell you how exciting it is to walk into this building and see the Philippine National Natural History Museum in this day and age and what a change that represents over the way things were 30 years ago. But what is a natural history museum, right? Is it really just a place for school groups to come and go and take students on field trips? Is it really just a place for, to celebrate, another museum to enjoy your time in Manila? Well, I guess what I would argue is that it's also a biodiversity library in a sense. If you want to think about the way libraries document scholarship and the writings of society, it's a record of biodiversity on Earth. And so natural history collections document both the raw science, the pure science approach, the basic and applied research of science, and that's what a lot of the scientists at the National Museum do. But also there's this human dimension, right? There's the way natural history collections document habitat loss and biological invasions, global climate change, and other things like rising sea levels. And of course, they address the information, the data that we need for public health and security, biosecurity for agriculture and other things that are immediately important to the human dimension here. And the other sort of intersection of big science and the use of natural history collections is ecology and conservation, which ties together basic applied research and habitat and conservation focuses. So if we think about those collections, like let's think about the ways in which those specimens are used, right? There's the data, the idea of an extended specimen beyond the specimen itself, but the way in which those collections are used. There's data integration and attribution, and by attribution I mean attributing those data to the people who collected them, the scientists like Ed Taylor and others before who came before us, but also from the local communities who have jurisdiction over those areas from which the biodiversity data were accumulated. We want to think about diversity and equity, inclusion, and access of all kinds of people to those kinds of data. And that is supported by the infrastructure and sustainability of those institutions that serve those data. But that requires digitization of those collections and serving them openly on the internet in an open access way, and involves workforce and education. That is a major advance in the way biodiversity data are used. And that brings us to 21st century collecting and what is the role of collecting biological specimens today, and how will museums be used in the future? And I think the sort of core agreement that museum biologists are coming together and agreeing upon is this idea of the extended specimen concept. The idea that there is the specimen, the single specimen that resides in collections, and from that specimen, various preparations or ancillary data streams are associated, like the morphology, the shape of the body of that specimen, photographs of it, CT scans, gut contents, tissues for use in DNA, molecular studies, hardcore genomics and use of the DNA that comes from those specimens, but also things like the information about how that specimen was collected, the date, the locality, the occurrence data, the habitats, species interactions that were detected at the time that collection was done. All of those come together into this idea of like, here's the specimen, and here's all the different preparations and all the different ways in which we collect and curate and take care of other information that derives from that specimen, the derivatives of that specimen, and how we serve it to the general public for everybody's use. So in the next section, I'd like to talk about this extended specimens and distributed databases. Now, here's the view of biodiversity collections, right, of big museums. Now, here's a view of the Smithsonian on the left. I think that might be Michigan at the top upper right. Here's the University of Kansas at the bottom right. Views of big collections that house specimens of dry collections in these cases. Many museums look like wet collections, involve specimens in jars of fluid preservative like ethanol, and that's what the view of the National Museum basement picture that I showed you from 2015 was, that a fluid collection. Some collections, like right now, most of the genetic samples for mammals, birds, and amphibians and reptiles from the Philippines are residing in long-term cryopreservation dewars in the basement of the University of of Kansas Biodiversity Institute in liquid nitrogen, and we built this this shelter at the base of in the basement of the museum that's impervious to natural disasters, and come up with a system of preserving genetic material in ways in which, if there were tornadoes or other natural history, or sorry, not natural disasters, that these specimens would be safe and preserved for the future. Because when we look at this view of how specimens were 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 sort of at risk over history, over history and over conflicts in World War II versus natural disasters, in some of the ways in which Philippine biodiversity has been documented in the past, there's concerns for all those different preparations, right? There's the specimens, these ones right here, but then there's all the data associated with them. And if those data are just sitting in a notebook, a ledger in the basement of the Natural History Museum, and a flood comes in and floods the bottom two floors, all that information is lost. And it's already been lost once in a big way, right? In World War II, when the Japanese bombed Manila, we lost all the information for all the species that had been recorded up to date. That's about 400 species of amphibians and reptiles, including 30 to 40 original collections, the holotype specimens. We don't want to take the chance of that happening again. So this idea of redundant collections or numerous specimen preparation types being distributed across multiple institutions or different preservation types is really attractive, because it's a way to protect them from inclement weather, natural disasters, political conflicts, whatever, so that these data do not get erased if there's a disaster. And what has happened in the past is that lots of specimens and different preparation types have been preserved in different museums. So like the specimens might be housed at the National Museum of the Philippines, but parasites that came from them, or tissues that came from them, or recordings of the mating calls of those species of frogs or birds, or the genetic material or the photographs of them in life, those have all been housed in different places. And the idea with the extended specimen concept is to link them all together and have all those parts come back to the original specimen, and have them all be accessible to everybody who wants to access them, whether they're scientists or students or educators or policy developers or conservation biologists or wildlife managers, to be able to find everything and to not have to ask, but be able to find that information online and self-serve and find the information you want. So in the past, a specimen might look like this. This is a specimen at the University of Kansas. This specimen, this label tells you when. It tells you what it is, Atelopus petersi, where it was from, Ecuador, when it was collected in 1959 or something. And there's one specimen and 3 pieces of information. But there's really so much more information that we want to make, that is preserved and has been housed in different places because of the different nature of those digital, or the different nature of those ancillary information streams. Tissues, live photographs, preserved photographs, CT scans, digital x-rays, pictures of the habitat, recordings or sonograms of the mating calls, other things that we can't even imagine, field notes, et cetera, et cetera. And here's the thing. When it comes to biodiversity collections, the what, the where, the when, they just provide a baseline for detecting change over time and space, if we think about it this way. And I'm going to ask you to bear with me here. Natural history collections have the possibility, if we think of the specimen as the phenotype, and we think about the derivatives of it being the genotype, its genetic material, and the environment from which it came, natural history collections have the opportunity, or present a potential, for us to consider viewing change over time, right? And so in this instance, we're talking about the possibility of, if we have specimens from the same place from 1900 and from 2000, we might be able to compare what has happened to the species, to its environment, to the other species that are associated with it, to the community that it lives in, through time. And so keep this in mind. I'll come back to this point when we talk about a recent paper that does that, a century later comparison. And it's the natural history collections, these right here, this gray shaded thing, that provide the opportunity for us to make temporal collection comparisons between past and present or future scenarios. So when working in a collection, what does one do? One goes through the process of accumulating specimens. There are all of our field work. We preserve the specimens, bring them to the museum, take good care of them to the best of our ability, and then we share that information. In the past, we've done this sharing by entertaining visits from scientists, from professional visitors who come to our museum, or sending specimen loans out to other researchers so they can look at the specimens and measure them themselves. We respond to queries by researchers, and in the old day that meant like the written letter, or nowadays email or something else. We respond frequently to the general public when we get questions from educators or whoever about species in their domain, and we do this most at this point in time mostly through digitization of these specimens and their related information. And here's the point where, for transparency's sake, I want to make this point really clear. This is my memorandum of agreement between the University of Kansas and the central government of the Philippines, the Department of Environment and Natural Resources. And all of these specimens that are now housed at the National Museum of the Philippines or at Father Saturnino Urias University or the University of Kansas all come from the same collections because we're all working in collaboration. But I commit with this memorandum of agreement to commit that duplicate samples of the specimens shall be deposited in the Philippine government through the National Museum in the Philippines. All the holotypes, that is the original collections, the first specimens of each species, for example, they go to the National Museum of the Philippines. And all of these different research possibilities are possible, provided that ownership of those specimens shall remain with the Philippine government. So these are your specimens, and we may be taking care of them now, but they can be recalled by the Philippine government at any time and brought back to the Philippines. And in the meantime, those specimens and their associated data need to be made available to the user community, and that's conservation biologists, educators, students, everyone who wants them. Because again, we want not just the specimen, but all this information associated with it, all the other information that was collected at the same time as the specimen should all be linked together and be available, open access to anyone who wants to use for it. So I sort of think of digitization process as kind of like a 1.0. Digitization 1.0 was imaging the specimens and taking, some primary information about them, conversion of specimen data into digital content, like scanning field notes, scanning old transparencies of photographs, taking this information, maybe using AI to transcribe somebody's handwritten field notes, getting all that information that was in analog form into digital form. And then we distribute it through a relational database, in this case, Specify. So here's a specimen of Microclamius stavinkii. There's a specimen. Here's a digitized photograph. All the information is served in a public way that can be accessed through the internet. And it's all accessed through the Global Biodiversity Information Facility it's the IDIG-Bio, the U.S. Aggregator for information, and potentially through individual institutional portals. There we are, right? That's one way to share biodiversity data through these biodiversity information products like GBIF, IDIG-Bio, and KU Herpetology, or databases associated with other museums if they have them. Now, I just want to give a couple examples. I'm going to walk you through just everyone through one example. This first one is just a discovery of a new species of lizard that is this beautiful, really extraordinary lizard, and describing it was an interesting experience for us because it was such a distinct new species of lizard. And we described it together with some collaborators from Partido State University and Ateneo de Naga University in Naga City. This is a species of pseudogecko, which is a really rare and hard to find species of gecko that is very secretive and lives in the forest. And we sequenced genetic variation from it and showed that pseudogecko hongkog, which is the new species, was genetically distinct from all the other described species and falls out in this clade all by itself. And so we derived the species name in Tagalog using the Tagalog term hongkog, which means hollow or pocket or empty, in reference to the fact that this new species likes to live in cavities and in branches and live in hollow areas, but also in reference to this problem of all these gaps in biodiversity distribution data. In particular, protected areas often turn out to be areas that have no records for occurrences of species of interest in them, because protected area management boards tend to try to keep out hunters and everyone else that might harm the biodiversity there. And so the end result is a artifact, which is the protected areas, ironically, turn out to be places where we have no information about the biodiversity. So we called it Hunkog as a result. So we're serving the data for pseudogecko Hunkog through the specified database here at KU. And you can see the paratypes and the holotype. And if you want to learn more about it, you can click on the holotype. You can get all the information for that holotype, which is now National Museum of the Philippines catalog number 9864. Here's all the information that was collected, the occurrences, the georeference locality for it, etc. Now, this all teaches me like three really important lessons before we do field work. And I hadn't thought about this until this experience of describing this species and giving it a new name. Three things that we all really need to think about before we do these kinds of field work and share this kind of data. It's really important before we go in the field to think in advance and to identify the partnering institution that will accept, that will curate, and that will catalog your specimens. It's not okay to just go and collect specimens and bring them back to your lab or bring them back to the NGO office and put them in a cabinet and hope that they'll be okay, right? We really need to think very carefully about who will take care of these specimens and make sure that they're there in 50 years or 100 years, and who has the resources to make sure that the specimens, however they're preserved, will continue to be in good shape, will be taken care of, will be cared for. Before fieldwork, it's really critical, likewise, to identify the institution that's capable of freely sharing your data. If that's your intention, to make your data available for future generations, we need to think very carefully about what institution has the capacity to share data and to do it in an open access way. And so I think thinking before we go in the field to identify the partners, to identify the institution that can do it, that has the facilities, that has the technology, that has the funding for the care for the specimen and the way in which the specimen data are shared, it's really a primary consideration. And then finally, make concrete plans for sharing all the related different forms of data streams. Whether, in this case, here's pseudogecko Hunkog, which we've posted to GenBank. Here's the original citation. Here's, again, the holotype 9864, National Museum of the Philippines. Here's where we posted the data so anybody can download the sequence data for this species of gecko. And if you don't agree with me that this should be recognized as a new species, that's fine. You can download the data and do the analysis again yourself. So this is not an exercise in convincing me. If someone disagrees with me, it's perfectly fine for them to go get the data and redo the analysis. And I mean that for habitat photographs, field notes, photographs of the animals in life, DNA, derivatives, or any other biodiversity information product that we get downstream from our research that involves the specimen. All of these products, all of these different forms of information that come from that original specimen, making them all available, open access online for free and accessible use by all. Okay, so I mentioned that's sort of digitization 1.0, but 2.0 involves a much more advanced form of products. And we're going to hear more about that very soon. Photogrammetry is a great example. 3D mapping or modeling is another. You're going to hear a lot about this in Dr. Duncan Urshick's presentation coming up here real soon. But it's possible that we could digitally share 3D rendering of the specimens themselves, and that way people wouldn't have to come visit our collections, and people wouldn't have to come borrow specimens from us. They might be able to download them in this form and work with them directly for whatever taxonomic, education, conservation related source. I want to wrap up and talk about scientific transparency and inclusion and reproducibility, and I'll do that by talking about this one publication that my students and I published just recently that looks back over the last 20 years of Philippine herpetology and thinking ahead towards an increasingly collaborative, equitable, inclusive approach to the study of amphibians and reptiles. In this paper, we showed a great increase in the numbers of publications on amphibians and reptiles in the Philippines over the last 20 years, from 2002 to 2022. That's all really good news. Super upward trends of publications and people studying and publishing on Philippine biodiversity. And we showed what topics were published on frequently and which ones were less frequent across amphibians, lizards, snakes, turtles, etc. These numbers, these proportions haven't changed much in the last 20 years. But we were able to sort of revisit an earlier paper from 20 years ago that addressed the same questions. But we identified some gaps that were things that we need to work on. We all need to work on as a community. One is that we take the gender of the first author in these publications as The first author we think of as the person who led the research, who was the leader of that research. And in most of the publications over the last, well, okay, the good news is that there are an increasing number of women in Philippine herpetology, which is great. That spike there gives me a lot of hope. The part where we need to work on is that there are many more men than there are women who are publishing on Philippine herpetology. And that's something that I consider to be a challenge and a gap that hopefully we can all ameliorate in the future. The other one is that there are more foreign biologists publishing on Philippine herpetology than there are Filipino-led collaborations publishing on Philippine herpetology. That's another gap that I hope we can close in the next 20 years. The good news is that if you look at the relationship between the number of species and the year that they were described, new species or species from the Philippines, the up and from all the way from the sort of era of discovery of early 1700s and 1800s, all the way up until about 1960, most of the publications on new species of reptiles and amphibians in the Philippines were foreigners. After about 1965, 1967, most of the author lines of those papers involved Filipinos. So I'm increasingly comforted by and encouraged by this fact that Filipinos are describing their own biodiversity. In this paper, we also tried to identify gaps in the geographical coverage. We focused on where species, what kinds of studies were done, and what kinds of publications were involved. But here, in particular, I wanted to draw attention to a couple key areas where we have very little information over the last 100 years about about what the communities of amphibians and reptiles from these particular areas. Those represent challenges for the future, and I hope calling attention to them means that people will focus on biodiversity research in those areas. Last, I want to talk about integrity and repeatability of Caraga region science and maybe the impact that it might have on biodiversity conservation policy in Caraga. And I want to come back to this publication that has just come out that I alluded to earlier, where Doc Tess and our collaborators and I went back to consider the amphibian and reptile biodiversity of Agusan Marsh. Now, Agusan is right near where Ed Taylor, 100 years ago, spent a year and a half. This is a trained herpetologist, and he described about 100 species of amphibians and reptiles from the Agusan Basin and described it as this incredibly diverse area. Over our revisit, right before the pandemic of this area, we found about half as many species. So about 50% of Taylor's original estimate. That could be alarming, and it could be scary to consider that it's possible that so many of those species have disappeared. And in fact, that's what we wanted to focus on was, are there species that have appeared since Dr. Taylor did his original studies from 100 years ago? He was there in 1912 through 1915, I believe. If you're interested in that stuff, there's a book called Recollections of a Herpetologist that I recommend people take a look at. So we're interested in species that had appeared since his time, and also species that we couldn't detect that he had recorded and that were not available when we went, or weren't present when we came back to do follow-up surveys. Now, we only found about half as many species, and we were really focused in the marsh itself and not the whole basin. So that could account for some of the differences. But if you look at these accumulation curves of cumulative numbers of species by the number of survey days involved, you can see that none of them fully asymptote and level off, which probably says to us that there needs to be a lot more work in that area before we can conclude that we have a reasonable estimate of the total diversity that's there. So the fact that we only got half of Taylor's diversity is not so alarming when we look at these curves, suggesting that there's a lot more work to be done. But in the process, we found a bunch of really interesting species, and in this process, we also made a very concerted effort to serve data for like some new species that have been, or species that are very rare and hadn't been seen since, like Lepinia semperi or Amoya ruficata, really, really interesting species. The journal that we publish in, Zokies, has a requirement of publishing all of the information, all the original biodiversity data associated with those 420 occurrences from our surveys there in Agusan Marsh, with taxon match, 100% with coordinates, 100% recorded by year. And when one does this, there's all sorts of information that is made available and that is curated and archived in a way. There's a study of the study extent, the sampling that was involved, the methods, the contacts for accessing the original data, the purpose, the geographic scope, the methods that are involved, it's all there so that it can be repeated in the future. Now, if you had spent a lot of time working on a project like this, wouldn't you want all that information to be available so people could compare their future data to the data that you collected, right? I mean, I think like this was a great. I was impressed with the way in which the way in which the biodiversity information product of GBIF, the Global Biodiversity Information Facility, made this available, and DocTest handled all the curation of all these data. But if you look at the taxonomic scope, you can look at all the taxa that were recorded in our survey. Let's say you're interested in one, click on it, and it gives you all the original data of all the species that were there. You don't have to you don't have to digitize our paper or contact us to ask for anything. You can just download the data and reanalyze the data yourself. Pretty neat, right? Well, this is important because in an effort to round up all the biodiversity occurrence data for amphibians and reptiles in the Philippines recently, we downloaded all the global biodiversity information facility data and curated those data so that all the information about current biodiversity of Philippine amphibians and reptiles are up to date. And that results in about 70,000 specimens. We looked at iNaturalist and other citizen science sites that might add about 4,000 occurrences to that estimate, and a couple more thousand from material citations. But of course, what we want to know as biodiversity specialists is where is the biodiversity and how well does it align with these key biodiversity areas, or how well does it align with protected areas? Here you are at Mount Hilong. There's the occurrences, basically 70 species from Doc Tess's collections here from Father Setterner Urios University right there. And here's all the data for the rest of the Philippines. This is work that's under revision by a paper that my current doctoral student, Kier Patogo, has in press. And we've looked at all the sources of data. We've looked at the original specimen-based data. We've looked at citizen science data and material citations. And you can see there's a lot of gaps, right? There are lots of places in the Philippines where there are large, substantial gaps. Southern Mindoro, Eastern Panay, central part of Mindanao here, northern Samar, big, huge gaps that should be the targets for future biodiversity surveys. We suggested a couple ways to enable the mechanism of adding data to this and adding primary biodiversity documentation. So hopefully this paper will be accepted soon and we'll have this information available. But I want to focus on mainstream biodiversity documentation, promoting those institutional connections and institutional support for biodiversity data, and developing robust data management infrastructure. Why is that important? Well, before we go in the field, and this is our recommendation, this is a paper that came out in PNAS a couple of years ago with Doc Tess and me, and a large collaboration of people from many institutions around the world. We sort of came together with a set of recommendations for how we hope that people can interact in biodiversity surveys in the future. And so this set of equitable principles for fieldwork in biology led us to come up with 4 general principles of urging people to be collaborative, urging people to be respectful, urging field biologists to be 100% legal, to follow all permitting requirements and restrictions, and then, of course, to be safe. This is all important because before people go in the field, just like anything else, we want people to develop plans so that they're safe and legal and collaborative and supportive. But just like when you go camping, you think of all the supplies that you're going to need, your headlamp and your chair and your tent and all the things. We'd like to urge as many biodiversity field biologists to also think about, where are your specimens going to go? How are those specimens going to be cared for so that they'll be available in 50 years or 100 years? Who's going to take care of the field notes that you take when you write in your notebook? Just like everything else, plan in advance to care for, curate, and share your data for future generations. Who is it that's going to take care of the recordings of frogs that you recall, or for your photographs? Is there an online digital archive where you can send your photographs or anything else? All of those things should be part of the planning before fieldwork and before biodiversity studies, just like making your list of your clothes and your supplies that would be for a camping trip. So those are our recommendations, because, and I just really want to tell you from my own experience, I've learned so much from talking to older members of the communities that live around our field sites. I've learned so much from interrogating them, from interviewing them and taking down their memories and their recollections of where the forest used to be, what species used to be common or what species are not. I've learned so much from thinking about biodiversity across time, and that has involved me interviewing and talking about it and recording the information and finding a way to archive that information from the last generation. And in the same way, I want to make the data and the information, the stuff that I care about, available to the next generation. We have a lot to learn from each other. In a big field expedition like this one in Caramoan a couple of years ago, I learned a lot from all these participants in this field work. And on these regular trips, the part of the experience is part of the process. In addition to the data that we gather, we can learn a lot from each other and try to make all of that available to the next generation. So I'd like to just wrap up by thanking the members of the herpetology division here at KU who've sort of provided so much support for my research, a bunch of my students in my lab and members of the Biodiversity Institute. And I want to thank all of you there at Father Saturnus Urios University, in particular, Father Randy, Father John, VP Arlen Floretta, and all of you for coming today. And thanks so much for the invitation to present this research. Thanks very much.
Okay, Dr. Brown, on behalf of the entire Congress, we extend our deepest gratitude for your remarkable presentation. So please do hang on, Dr. Brown, on while we proceed for the next speaker. Thank you, Dr. Brown. Our second plenary speaker brings to this Congress a perspective that is nothing short of revolutionary, one where cutting-edge digital technologies meets the ancient diversity of living forms. He's a professor and director of Digital Life Initiative at the University of Massachusetts Amherst. Under his leadership, Digital Life has pioneered the use of photogrammetry and 3D imaging technologies to create stunningly accurate and it's really accessible digital models of Earth's organisms, and is essentially building a living digital museum of life on our planet. His work seeks the dynamic intersections of functional morphology, evolutionary biology, and digital science, asking not just what organisms look like, but how their physical form has evolved in response to life demands, how we can preserve that knowledge in digital form for generations to come. This morning, he will speak to us on the application of 3D imaging in functional morphology and evolutionary research. This morning, you will also witness the biodiversity documentation in a way you may never have imagined. Please join me in welcoming a professor and director of the Digital Life at the University of Massachusetts Amherst, Dr. Duncan J. Urshik.
It's a great honor to be here, and it means a lot to me to be speaking with all of you. And hopefully I can come and visit you soon and meet many of you. I'm going to share my screen here. As was just mentioned, I'm going to be talking about the work that we've been doing over the last 10 years or so, really building a new future for digital specimens through live scanning of animals. And as this image of a live blue whale indicates, our concepts about what natural history collections represent ultimately are grounded in all technology, the technology of the day that continues to evolve. Obviously, we'd love to have a blue whale in a museum, and we do have blue whales in museums. We have their bones, we have their tissue, but we also what does a blue whale really look like? How do we know? Actually, it turns out there's a lot we don't know about these animals, and there's a lot we have to learn. And that's what this project is about. It's about opening doors and opening minds about new ways of thinking about the world that is around us and using those as tools for natural history and for collections and for science. And the premise of all this work that I'm going to be talking about is 3D scanning. And you can think essentially of 3D scanning as a form of digital heritage. Most of you are going to be familiar with CT scanning, unless you work specifically in a field. When you have something bad and you go to the doctor, and has to be a CT scan or MRI scan of some body part that looks suspicious. And these technologies are fascinating because they continue to evolve. When CT scanning, MRI scanning first was created many years ago, the output was very different than what we see today. Today, you can get a beautiful 3D image, such as this one you see here, of nervous tissue. But previously, all you could get was really images, single slices. It's only today, many years later, that we can take these slices and create very elaborate 3D models to re-understand many of the structures that we look at. Also, many other forms of scanning exist that can allow us to understand not just biology, but also human history and architecture and art, such as laser and white light scanning that has been done on ancient buildings, as shown here in the bottom, which are buildings found in the Middle East. As the war currently in Iran has shown, everything in this world is ephemeral, especially buildings, artifacts, history. And when we take a physical shape and we scan it, we're not replicating it. All we're doing is we're creating a historical record that can hopefully be disseminated and shared. And finally, a technique that is really growing, it's one that we've used a lot, called 3D photogrammetry. 3D photogrammetry involves taking a series of images or video of an object, in this case a toad, a live toad, and converting those 2D images to a 3D mesh and a 3D model. And this is a very powerful technique because it can occur at many different scales, ranging from something as small as a plankton to as large as a blue whale, or even larger. And this technology has been wide applications for biology, art, history, culture, forensics, and many other aspects. So some of the key advantages of 3D scans include that they provide a 3D shape map of an object or organism. When we think about what an organism is, and we can begin with external features of an organism, which is what our eye sees. It's certainly not the only part of an animal that's most relevant. Obviously, there's their internal anatomy, musculature, nerves, and so forth. But it's a key part. And as I'm going to discuss, there's certain key animals that we work with, such as sharks and marine mammals, that actually are quite hard to measure. It's very hard to capture a shark or a sea turtle or a marine mammal and do measurements on them. Natural history collections don't really have these kinds of specimens as well-preserved individuals. I'm not going to be talking about today, but in the future, one big part of our lab has also been looking at 3D scans as a key way to look at the color of organisms. Where they can also be modified to add movement, as I'll be demonstrating. And the end result can be strikingly realistic. And this goes to the point of when we work with animals in the field, one of our goals is to replicate not just their physical structure, but what they look like at the time of capture. This is, in essence, an ephemeral entity. When you capture a fish and it's preserved, this color changes, everything changes. And these results can be 3D printed and shared, much as Dr. Rafe Brown talked about. So all these are key advantages, but what about scanning living animals? When we think about certain key species that are on this planet, and above I've given 3 examples. We have sea turtles, marine mammals, and sharks. All of these are very critically endangered species. The vast bulk of sharks are very endangered, and that's true for these other 2 groups as well. These are species that are hard to work with in the field. Some of them can be very dangerous. Some of them are elusive. Some of them are very rare. There are often very strict laws protecting their acquisition and use. And so what does a sea turtle look like? What does a shark look like? It's a good question. And we know that certain species are very poorly represented in natural history collections, including many of the ones above. Yes, there are many sharks in natural history collections, but these won't be necessarily fully adult specimens, or they may be specimens that were collected over 100 years ago and are lying kind of crammed in a jar. There are many aspects that we don't know about these animals, the shape, their morphology, their life history. And as you can see in this case of a southern toad that was scanned at the University of Florida, the difference between a live animal and dead animal can be in some cases very subtle, in some cases quite extreme. And actually, we don't always know quite what those differences would represent. So this is one of our big challenges, to reconstruct what living animals look like. And yet it's a very big technical challenge. And this raises the question, what could you do if you had exact animal avatars? If you could see a rhino or an elephant or a frog or a lizard, and you could create a digital replica. And as I'll be talking later on, we're actually doing this not only with the exteriors of animals, but also the interiors, bones, musculature, and many other features. What could you do? It's a great question. It's a question I pose to a lot of people as I give these talks around the world. And I'll give you some of the answers that I often hear from people. But before I do that, I'll start with this story. This is Mac. Mac is a leatherback sea turtle that we worked with the Loggerhead Midlife Center and in Juneau, Florida. In this area of Florida, these sea turtles are nesters. They come out at night in June and July during the warm summer months. And it's really a sight to see, to go on the beach late at night and see an animal that can be up to 3 meters long, digging a hole and laying a nest. But these animals are critically endangered. They are actually among the rarest and most significantly persecuted of all sea turtles. They are truly open ocean travelers. There's a lot that we don't know about them. This particular female was interesting because she was a daytime nester. Normally they nest at night, but sometimes they linger on to the morning. This allowed the group that we worked with to capture photography and video of these animals. And actually, we were able to create an accurate 3D scan. And you can see all the nuance of what a sea turtle represents, the abrasions, the color, the texture, and the movement, which we reconstructed that is not really so much scientific, is more a representation of what a typical movement of the potential of what this animal could be. But here's some of the possible uses of digital specimens. You can collect 3D models of organisms in the field with DNA and release them. Many of the places I work with, and Dr. Brown and many of the others that you're familiar, many of you also work in, it's very challenging to collect animals. It's hard to get permits, and for good reason. There's a long history of exploitation of these countries by many scientists and other entities. So many of the areas, I'll name a few: Tanzania, Ecuador, Borneo, many other countries in the world are areas where it's very challenging to work with animals in the sense of getting permits to take animals to collect them. You can perform computational fluid dynamics on 3D animals of flying and swimming animals, just as I did with the leatherback sea turtle. You can rig them and use them to understand visualized locomotion. We are doing this as we speak with key animals such as African elephants, polar bears. These are very dangerous animals that are very hard to work with in zoos or in the field. They're hard to visualize. There's a lot we don't know about their locomotion. There's tremendous mysteries about them, and we can use them to understand them. We can also use them, as I'll discuss later, to estimate body condition, the health of animals, one of the most important aspects for any species, especially megafauna that's critically endangered in an era of climate change. And that we can use them, and many of our models do. They get used all around the world to design new kinds of robots that are going to be really the vanguards of many kinds of exploration we do in this world. And finally, I think not insignificantly, the work that we do, as I'll discuss, and 3D models of animals play a really important role in inspiring future generations. I was that kid that loved snakes, and if I could have had a great 3D model of a snake, it would have been awesome. And I know many young people reach out to me from all around the world, enthusiasts, and just talk about how great it is that they can access these animals through these free collections that we've created. And so the collection I'll be talking about is the Digital Life Project. The Digital Life Project is a open access project at the University of Massachusetts, where I'm a professor. And our goal is to recreate living animals in 3D and to provide those for free to the general public for nonprofit usage in conservation, science, and education. Our work has only just begun. We've only done about 120 3D models of animals. That's not very impressive. There's much work that needs to be done. But I will argue that what we have done is, instead of picking out the easiest animals to do, we picked out the hardest. Because our point of view is that if we could pioneer a wide range of different ways of doing different animals, and we could show other people how to do it, it would set the stage for others to do the same. So we've done many animals such as the southern white rhino, sharks, and sea turtles, and many others. With the 120 animal models, we've had over 1.3 million views and over 300,000 downloads. To me, what this says is the passion and interest in for life on this planet cannot be underestimated, and has to be. I'm always amazed at the passion people have for sharks and sea turtles, and that gives me hope. That gives me a lot of hope. So what I just want to finish with the last 2/3 of my talk is outline a few goals. The goals of the Digital Life Project are to provide the methods and 3D models to scientists and public as open access tools, to recreate the shapes of living animals in 3D, and to use these 3D data, particularly as conservation tools for endangered megafauna. I believe this is a very important task because it's important that we understand where these animals are going because they impact many other aspects of our environment. So I'm going to be talking briefly about the methods, how we recreate the shape of animals, and then how that relates to the conservation of key species. I'm going to start with one tool that we use. We use many tools. We use laser scanning, CT scans. And we also use a technique called photogrammetry. I thought I'd introduce it to you. In this technique, you take a series of photographs of an object. In this case, a golden poison frog, one of the deadliest frogs on this planet. This frog has these tadpoles on its back. It's a male frog who's taking care of his young. And through these series of photographs, we can take what is a 2D image and convert it to a 3D mesh, and ultimately lay a color map on top of that 3D mesh. So it represents almost like a cake. We have a foundation of the cake, and then we have frosting that we lay on that is essentially a mathematical cake that is laid on as a series of data matrices. This technology is powerful because of its flexibility. It can be done at many different scales. And one of the key things that we've done, and I'm glad Dr. Tess Sanguea has followed up on this in her amazing group, is some of the multi-camera rigs that we originally built, and we did take some of these to the Philippines a number of years ago, to scan animals such as small reptiles in the field. This is a 42 camera system, and this system was a system that we could use, and we did use to recreate many small reptiles and amphibians. But let me talk about two other larger species we did, because they were allowed us to really think deeply about how we can use these methods to recreate some animals that are very critically endangered. And those two species I'm going to talk about here are the southern white rhino and the great hammerhead shark. The southern white rhino is a species whose numbers have declined precipitously within Africa. And this decline has begun, actually dates back all the way to Roman times. The main reason for this is the tusk, or I'm sorry, the horn. These horns are greatly prized, and a horn of a rhino is worth almost $300,000 to $400,000 US because it is used in wildlife trafficking throughout many parts of the world. The other species, the great hammerhead shark, these populations have declined about 99%. Historically speaking, the largest great hammerhead shark ever captured was almost 20 feet long, or about 7 meters long. The largest species, this largest individual you'll see today is about half that size. It shows you the kind of persecution these animals have faced. But scanning them is no easy task. In the case of the southern white rhino, we first began working with the captive animal, this animal Bakari at the Perth Zoo. Rhinos in the wild are notoriously grouchy. Rhinos in captivity become very tame, and that was true of Bakari. We used a 20 camera system that surrounded the southern white rhino, and the Perth Zoo was very accommodating, and we were able to use that to recreate a 3D mesh, and finally a 3D render of color of this rhino. Then working with the zoo staff, we were able to verify a series of measurements of the rhino, such as the back length and the body length and other parts of the rhino. And the zoo staff also took images of these animals, and it really allowed us to understand the morphology and confirm the morphology in a good way. This is the final render, and we were very proud of it because it really showed to us that we could recreate a very complex animal at a high level fidelity. And this model, by the way, is free and open access to use. And it has been downloaded over 18,000 times, which is very exciting to us that it would be used so frequently. A key advantage of 3D modeling is accurate estimation of volume and surface area. We stand a very important part in history. For the first time in human history, with these methods, we can actually recreate the volume of an animal. Unless you want to take a rhino and dip it in water, it's not going to be easy. In this case, this male southern white rhino we knew its mass, we knew its length, and we were able to therefore recreate its volume and its surface area. So with this technology, this is key set of mathematical variables that we could use and compare to other animals. Now on to the great hammerhead shark. These sharks, we knew that we could study them and dive with them and film them in an area of the Bimini Island in the Bahamas. In the area, the water is very clear. These sharks, you can swim with them. They don't tend to be aggressive. And this is the setup that we used. We had a series of cameras that we used with a photographer named Casey Sapp. And we used a person who fed the sharks named Sean Williams. That's me in the back. There's a current going, which the sharks follow to the food. And we use these cameras to recreate a 3D model of one of the key sharks, a shark named Nemesis, about a 3.5-meter female. And that shark is shown here. We were able to recreate this shark and ultimately recreate an accurate 3D model of what it looks like. And just like the rhino, this animal is available online for free and has been downloaded many thousands of times. The value of these scaled 3D models are, and all of our 3D models are actually scaled to size, which is important because when many people download 3D models, they download them from many different sources, but they're not at the same level as resolution. They're not the same level of scale. So we have here the 16 different shark species that we've scanned, ranging from the largest shark in the world, the whale shark, to one of the small sharks in the world, one of the pygmy sharks, and many of the sharks in between, great white sharks, hammerheads, reef sharks, and so forth. And we can, by looking at this image, it really gives you a sense for the variation size and morphology of these animals. The interesting part is that when you actually compare some of the scans that we did, this was some work from a former honor student did, Asana Seth. This is a comparison of great hammerhead sharks, sea turtles, and marine mammals. And we're plotting volume in the y-axis for surface area in the x-axis. This does not take into account the confound effects of phylogeny, but it shows you is that these, if we simply imagine that this is an isometric relationship, and the isometric slope will be 1.5, the actual slope is 1.48, and the R squared is 0.99. It shows you that despite the fact that we're comparing reptiles to fish to mammals, there is a basic relationship between these variables within the open oceans. And this goes all the way from a blue whale to a tiny hawksbill sea turtle. And it means that despite vast differences in body shape, ecology, and life history, animals from a lizard to a whale share the same basic relationship among mass, surface area, and volume. This is work, by the way, we have not published, and we're looking forward to expanding. The final part of my talk, I just want to spend a few slides to talk about the implications of this work that we do for conservation. It turns out that estimating the volume of an animal is a very powerful thing. It allows us to develop a series of methods to quantify body condition, also known as body mass. Among humans, usually gaining weight is not a good thing, and we tend to be obsessed with losing a few pounds, but that's not true for wild animals. For a wild animal, a fat bear is a healthy bear. A fat impala is a healthy impala. And that dictates almost everything about their lives, their ability to reproduce, their ability to survive, their life history, and their ecology. And so therefore, it's very important to accurately estimate body condition in animals, but it's very challenging. And there's an assumption here. The assumption is volume equals mass. As I'll show you later, I think it's a reasonable assumption. And so that raises the question, can we use these techniques to estimate remotely the volume of a whale or a polar bear or an impala or many other animals? Here is the ideal workflow. We have a 2D image of a polar bear. Maybe it's from a drone or a plane, or maybe it's from a car. We then use computer science techniques to estimate a 3D model. It has a volume, and then we use that to estimate a mass. But there are some assumptions here. We want to be able to relate volume to mass. We want to relate the 2D images of animals to volume. If we can establish those relationships, we might be in really good shape. And so we set out to prove that concept, and we did that. We had the opportunity to do that with impala, which is an African ungulate, a very common species in Africa. We worked with Kapindi, which is a female from the Fresno Zoo. That's her on the left. We were able to get a good scan of her using a series of cameras. This is Jordan Amato, was a student that we worked with on this project. We also built an internal skeleton of this impala, and that allowed us to understand as weight is gained and lost on the animal, how it affected body condition. We knew the mass and the length of this animal, and that was one of our key steps. And by the way, this work was also in collaboration with Matt Hutchinson. We use then in software a shape keys modeling approach that allowed us to correlate 2D images to 3D metrics. The most common way you'd access an impala in the wild would be either through a drone or a plane or a helicopter or a car. In the case of a car, we might take an image from the side, which is not that hard to do, or we might take with a drone an image from the top. Can those 2D images tell us about the volume of the animal? Because we have a 3D model, we could then recreate a series of either weight gain or weight loss with impala. In this case, we created 25 total impalas ranging from extremely emaciated to extremely obese. Obviously, the most obese and emaciated animals are not the most realistic. And then we can relate these images and the lateral and dorsal areas to surface area and volume. And this is those 25 points. And this is where the data get really interesting. If you look on the bottom part, we have volume on the y-axis and lateral area and dorsal area, the sum of them on the x-axis. And that r-squared is 0.99, which means if you take a lateral image of an impala and a dorsal image with these modeling approaches, you can predict volume almost to perfection, which is very interesting. And the same is very much true with surface area. Now, the big question is, can we relate mass to volume? We haven't yet done that within impala, but I'll show you a data set we developed with sunfish as a proof of concept from other data. This was a work by Olivia Sanborn, an undergraduate in my lab. She scanned 30 sunfish. We have mass on the x-axis and volume on the y-axis. And from those 30 scans, she did weigh all of them. The r-squared is 1, and the slope is 1. So volume to mass are almost perfectly related. This is encouraging. We hope to get more data on this front. So I'll end with this, the point of discussion, the digital specimen, which is we have in this case 3 examples I gave you: a great hammerhead shark, maca sea turtle, a rhino. These are animals that you might encounter in the field, and they're not going to be animals you can collect. But can we use our technology to recreate them digitally and store them in natural history in collections, along with metadata, and allow people to access them? That's the question. As Dr. Rafe Brown and others have talked about here, museums represent one of our most powerful innovations and engines of intellectual thought, and they will continue to be so. I'm a big believer in museums. I've used them all my life. My interest is in working with museums to keep building them up and use these digital tools as powerful extra aspects of the extended specimen so we can continue to understand them and grow them, and actually keep adding to museums is one of my main interests. So I'm going to end there. I want to thank you, and especially want to thank Dr. Tessa Nguilla, many of the funding sources, and for those of you interested in accessing our models, which are free online, you can go to digitallife3d.org or sketchfab.com/digitallife3d. Thank you very much.
Dr. Ershik, thank you for that visionary and deeply inspiring presentation. And now, ladies and gentlemen, we are now open for the floor of open forum with both Dr. Rafe Brown and Dr. Duncan Ershik. This is now your opportunity to engage directly with 2 of the most innovative minds in biodiversity research today. So if you have questions, whether you are here in the hall or watching online, please prepare your questions briefly and clearly. On-site participants may approach the microphone while our online participants may submit their questions through the designated platform or be called upon. So let's begin. Who would like to open our discussion? Anyone would like to ask question? Okay, please. Our microphone is in the center.
Hello, my name is Kenneth James F. Gasparil. There's some concerns about like how does affecting of migratory birds, some other like say this, pigeons here in the Butuan, in our place in Butuan City. Does it affect mostly like local birds or like well-known like Maya well-known birds like Maya here? Because in our place like there seem to be like less like Maya bird like sightseeings on that and more affecting on migratory birds and such. Will that be like much of known this isn't a problem here in like when you try to research that, is it more of affecting for the biodiversity here?
For whom you address your concern or
From Sir Dr. Duncan.
okay, Dr. Duncan.
Um, Thank you. Could you kindly repeat the question?
There's some questions like I say like migratory birds here, rotating here. Some, for example, that Philippine Maya, like a small bird from Philippines. There's some problems with like small sightseeings of that. Wouldn't that be, like small conducting, especially if there's like a huge, whale sightseeing of other birds like a pigeon, and also like
Um,
a question for that. Also, like a question related to that, like invasive species like janitor fish, most not that later, like like a decade ago, and in Philippine, like invasive species affecting biodiversity. Some of that like frogs or like I said, a janitor fish. How would that affect the whole diversity and affect like Philippines as a whole? Like, wouldn't that be like a huge deducting on like wildlife, especially if
hmm.
there's like local citizen problems?
So that's a great question. I would actually defer to some of my colleagues working more intensively in the Philippines who could really, really answer that question. Um, Maybe some folks, people in the audience or other people in the panel might be the best people to answer that.
Thank you. I think Duncan, he was referring to asking a question on whether invasive species could affect endemic or natural populations. I think that's the
Oh,
point of the question. Okay, so I think we're ready for the
right,
next
right.
question.
Hello, good morning, everyone. I'm April from Caraga State University, and my first question is for Dr. Rafe Brown. So, the question is, how can we avoid overcollection in the field, especially with specimens for cryptic species?
Sure, that's a great question. You know, as biologists, I sort of feel like we're the people who care most about natural population, and so we have to trust our instincts when it comes to overcollecting. But the real answer to that is that when, just like you're ready to go to the field. Sorry, I'm getting a wicked echo. But getting ready to go to the field, it's incumbent upon the field biologist to prepare as much information as one possibly can before doing collections or collecting new species. So, the answer to your question in my mind is having free access to all of the world's collections from that area of that, so that people can make biologically informed decisions whether or not to collect a specimen. Because of course, there are times when we don't need to collect any more specimens. And there certainly are times when we need to collect. And if we don't have access to all the information around the world, including collections from Europe and collections from the National Museum of the Philippines and collections from University of Kansas and from Father Saturnine Urios University collections, if we don't have access to all that information, we go into the field handicapped, right? So I sort of feel like the best answer to your question is when is the best, how do we avoid over collecting or certainly redundantly collecting a population that has been collected before. We don't want to harm populations. And as biologists, we're the people who we care about populations more than anybody else, right? So I sort of feel like the answer to your question is the extended specimen, right? Is the access to distributed databases around the world. And before we go in the field, the best thing we can do is download all the biodiversity information for that area. If we're going to go collect in Kababaran, then we need to download all the information for the Mount Hilong Massif, get all the data from all the European collections, from the American collections, from the Philippine collections, Australian collections, bring all those data together and walk into the field perfectly informed about the state of the art of the global collections of biodiversity from that area so that we know when to leave specimens alone, to leave populations alone, and when we know it's critically important that we collect that specimen. So, yeah, thank you for
Thank you so
that
much for
question.
your
That's a great
answer.
question.
Okay, I have a follow-up question actually.
Sure.
This question is for Dr. Duncan. So in connection with my previous question, how can we digitalize those cryptic species?
That's a great question. I think it's very possible to do that. I think one of the things I should mention is that um, we haven't focused so much on doing volume of specimens. I think it'd be interesting to do much wider range of specimens, reptiles, amphibians, fish, birds. The reason we haven't done it is we haven't had a lot of funding to do it. We've really focused on more methods that we can do with a smaller number of species. And I really think it'd be a fantastic idea to scan a much wider range of Philippine biodiversity. Philippines is an amazing hotspot of biodiversity. There's so many amazing species there that if they could be shared with the outside world would be an incredible innovation. So I think it's simply a matter of um, investing time, resources. One of the things that we do is we do work with a lot of museums to try and train up students to do the best practices for all the work that we do, because the work we do is highly specialized and highly skilled. And I think that's one of the big limiting factors is training. We need to do more training of young scientists who are often very good with technology to do a lot of the work that we do. So I think Dr. Tess Sanguela is doing an amazing job scanning frogs. And I think we just need to expand more of that. And I think that there's a lot of hope for the future.
Thank you for that answer.
Some more questions? Okay. Please don't forget to state your name and from what school are you. Okay. And for whom you address your
So, good
Ar-
morning, everyone. I am Shirimi Favroa from Ateneo de Davao University, and I am addressing this question to Dr. Duncan. So, it is about the 3D modeling. So, this is all about the ethical standards or guidelines. Do we have a general guidelines or ethical standards when it comes to 3D modeling? And is it generally used for each country, or when we apply to our to our ethics committee in our university, is it easy for us to conduct this study using 3D modeling or photogrammetry when it comes to ethics? That's
Yeah, so that's a fantastic question. The one thing that's very interesting from a technical point of view is that a 3D model is really just a photo. So the same rules and laws that apply to photos, I believe, apply to 3D models currently in terms of, so in other words, if you were to go to Africa and take a photograph of an elephant, and then let's say you took many photos, which people do all the time on safari, and let's say you took 100 photos of an elephant, and then you create a 3D model of that. I, my view of that is that is essentially, as long as it's for nonprofit use, is entirely reasonable. I think that um, the only question would be, and obviously it falls into very non-invasive work. Um, the one thing I'll ask, I will, so I think, but I think you're right, that I think what's happening now is that a lot of countries that I work in are becoming increasingly interested in these 3D data, um, and are starting to put restrictions on them, which they're saying that, for example, they might own the photography that you take on an animal. And I think certainly every country's got to work out what's right. I guess the only argument I would make is that this is scientific data. And, obviously, if you go into a country and you just get a permit to take photographs and export 3D models, I think that's extremely non-invasive. I think the one standard that we do have ethically at the Digital Life Project is everything is open access. We actually always do get permits to do our work. For example, we worked in the Philippines or worked in Asia or Africa. We work with local partners, much as Dr. Brown elaborated, and we always make sure that our models cite that collaboration and those models are open access. So I think there are a set of ethical standards that we apply for, but I can tell you certainly some countries around the world are creating their own standards, and I think that'll be interesting to see how that's applied. That's a great question.
Hello everyone. I am Lloyd Ternat from Agusan del Sur State University, and my question is for Dr. Duncan. My question is, I have this YouTube channel that I always watch, and they uses phone cameras to like scan objects to use as 3D 3D assets for their indie films. Do you think the technology that you used for scanning organisms can be accessible, like having a phone, like a single phone to scan an object or an organism? If it can be accessible, it could be that accessible. How do you think it could compare to the quality of having like rigs and multiple cameras to capture the 3D model of an organism?
So that's a great question. I think people, photogrammetry with standard cell phone camera like a Samsung phone or a iPhone is often can be very high quality. You're looking at 12 megapixels or higher, standard, very good video resolution. So there's outstanding opportunities to do citizen science, and people have done quite a bit of it. I would actually argue that the innovation is not on the camera end, it is on the software end. And there's tremendous innovations in software. So I alluded to some of them. We know that in computer science world, there's tremendous innovation in what we call 2D to 3D projection. So you take a photograph of a human face, and there's enormous innovation in actually extrapolating that to a full three-dimensional human face based on data matrices of the way the human face is shaped. And so whereas camera technology is actually pretty well established, and so a cell phone is actually a very good usage. What is changing very dramatically is computer science and all the tools behind it. And that's actually one of the biggest areas we're growing into. So yes, I agree that a lot of what we did, I think, could be done by, and it actually is being done by everyday citizen scientists all around the world. And I certainly would have hoped that there'd be a lot more work done to recreating living animals all around the world and putting them online, and so we could build up a much bigger library. I'd say some of the limiting factors are some of the work that needs to be done in post-processing, that we work with in Blender and other software that still is highly skilled work. And that's again where I think innovation software is going to have a big difference. There's a lot of work that is kind of making that whole process much easier. So I think the next 5 years, you'll see tremendous innovation in all kinds of areas of model creation. And that's one of the reasons why, by the way, we haven't gone out and just tried to model everything. We think there's going to be ways to do it much more parsimoniously in the years to come. Thank you. That was a really great question.
Thank you.
Hello, hello. Good morning, everyone. My name is Rafael James Panal from the Philippine Normal University, Mindanao. And I would like to address my question to Dr. Brown. Um, about the collection of live specimens, do these specific specimens are being monitored after taking it? Because it was mentioned at the session that some species will change over time, mainly because of evolution or adaptation to its environment. So how can we assure that these animals that were taken are still the same and have evolved and not some kind of new species, sir?
That's a great question. Um, considering biodiversity through time is one of those challenging things that we may do as biologists. Considering the changes that might affect populations through time, is something we all struggle with, especially in museums where we have these ancient collections from 100 years ago or 200 years ago or 400 years ago. That's our challenge, is to think, can we use these collections to inform us about how humans have impacted the environment or how these species have evolved? That's a very short window of time in evolutionary sense. Um, and so I get, your question is a really good one. I don't really know the answer. Um, I think the best thing we can possibly do is document as much as we can, and that addresses or speaks to all the things we've talked about today in terms of all the different ways in which we document biodiversity. Um, because the one thing that we're sure of is that we'll use these specimens and their data in different ways in 100 years than we could ever have imagined. And so the best thing we can do is document the hell out of them now, put everything into archives so that it can be accessed in the future, put everything in digital format because no one wants to go back through old physical archives and libraries and try to recreate the digital world that we're in now, especially with AI and all the uses, the positive uses of those new emerging technologies. Best thing we can do is document them as much as we possibly can, so we can answer those types of questions in the future. I can think of many examples, but I don't know a hard answer to your question, but it's a great one. Yeah, thanks.
Good day, everyone. I have a question addressed to Dr. Brown. I'm Alexander Archibald from Father Santeriorios University. And my question is, how can aspiring field biologists tackle any kind of unexpected limitation presented in collecting their own data without jeopardizing their natural habitat? Factors such as the environment the researchers is exposed to the way the animal behaves, and other factors as such.
I think we all, as biologists, have the obligation and the responsibility of not impacting populations negatively. The thing that we usually do as herpetologists, people who study amphibians and reptiles and 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 thinking about the different ways in which amphibians respond to the time of year, right? The rainy season is when all the frogs come out. The dry season is when all the lizards and snakes come out and breed and when they're active. The thing we do is we go into the field site and we try to get an inventory of everything that's there, and then we try to see what's really rare and what is pretty common. And if we apply the same standardized survey approaches to sampling a transect with a little bit of time, 10 days, 12 days, 15, 21 days, we start to get a sense about which of these, which of the species in our list of everything that's present, which of the species are really common and abundant, and we see them every day. And so we do this process of daily lists where me and all my field assistants get together, six of us who know all these animals really well. We get together, and every single day, this is something bird people established early on, is making a daily list of every species that has been recorded that day. And so for us as frog biologists, for example, we're recording as present a species if we collected a specimen or if we heard it call, right? So even if we didn't get it, we didn't collect it, we know that it's there if we heard it in the background. So we get this daily list of all the species that are detected each and every day. And after 10 or 15 days, you can statistically you can use a couple different approaches. Statistically, you can start to determine whether you have sampled or detected all the species to there's two ways to do it, these kind of surveys. One is to sample to equivalent effort, and that's the way mammalogists, for example, in the Philippines have done things. They've gone out and they put out their 30 bat nets, and they register the numbers of bats that they recorded as a function of net nights per, as a function of number of nets per night. And they sample to equivalent effort. And so they do that for 2 weeks straight and have 30 nets. And then they do that at high elevation and low elevation, and they compare them statistically. The other way is to use the Chow estimator, a statistical approach, to try to estimate sampling to completion. And that's the way that we've been trying to do our surveys, which is to statistically on the fly estimate whether we think we are arriving at a near complete estimate of all the species that are there. And in that process, we can look back at the last 2 weeks and decide whether species are really common or whether we see them every single day. And so that's the way in which I think I personally make what I feel like are informed decisions about whether we need to collect a specimen and whether that will impact the population or not. Does that answer your question? Is that approach? I don't know.
Yes, Dr. Brown. Thank you, Dr. Brown.
Okay, thanks. That's a great question, and that's something that I think field biologists have to think about all the time, because we want to practice sound biology, and we want to not negatively impact populations. And if there's a better way to do it, then I want to hear about it. If there's other ways to approach this, let's have conversations about it, because we don't want to hurt populations, and we certainly don't want to jeopardize the future of individual species. But I think there are things that we can do as biologists in the field to determine whether we are having a negative impact on populations by collecting a few specimens. Thank you. Yeah, good question.
Good morning, everyone. I'm addressing this question to Dr. Brown, sir. I would like to raise this. Here in the Philippines, we have this online checklist or database for Philippine plants, native plants. We call it Digital Flora of the Philippines database, where contributors from all over the Philippines contribute photographs from their field works of the different plants that they have seen from their work. So my question is, are there any current large-scale work or efforts that are underway here in the Philippines that you know of, which aims to digitize animal specimens collected from field works which can be accessed by the public? And what steps or maybe tips or recommendations can you give to those who are starting or who wants to pursue such effort?
Also, a fantastic question, and one that really strikes me right at home. When I first came to the Philippines in 1990 as an undergraduate student, I spent a lot of time at the National Museum of the Philippines, and at that time, there was a big project called Flora of the Philippines, and I'm just going to show this picture again. Vicky Funk, this woman right here, was working together with a biologist named Ben Stone, and they were collaborating with members of the National Museum of the Philippines to produce a Flora of the Philippines, an entire botanical account of all the biodiversity of plants in the Philippines. That project made a lot of progress. There was a bunch of funding at the National Museum of the Philippines. I met some of my earliest field colleagues at that time when I was undergraduate who were working as part of that project. And then Ben Stone, who was I think he was at the Smithsonian, died of a heart attack in Manila, and that sort of ended that project. But I always thought about that over the years, because Vicky Funk and Ben Stone had this impact on me early on as a young undergraduate student, um, by challenging me to think about what would it take to establish uh, a comprehensive account of the entire amphibian fauna of the Philippines. Why would that be so hard? And I, at the time, as a 20-something-year-old student, I thought, okay, no, I'll never be able to do that. Um, uh, and to list all the species that occur in the Philippines because it seemed so overwhelming compared to where I was from and that boring, flat, homogeneous landscape that I grew up in. Um, but they were doing it with plants and doing it with the Smithsonian and through the National Museum of the Philippines. And they were getting started on that whole progress. And I just feel like that was like a big lesson to me, like dare to think, as broadly as as one possibly can, how much would it take for us to come up with a complete list of the fauna or flora of the Philippines, or complete lists, a photographic guide to all the frogs of the Philippines. What would it take to come up with a key to the snakes of the Philippines? How hard would that actually be? What would it take to develop a book on all the amphibians and reptiles of the Philippines, or all the freshwater Philippines fishes of the Philippines? And unless we ask ourselves those questions, and unless we have the courage to do what Dr. Urshik has done and start with blue whales at one end and Max the leatherback turtle and all the way down to smaller units at the other end, unless we like challenge ourselves to think about what the challenges are, we can't answer those questions. But I sort of feel like if we think as broadly as we possibly can, um, the answers may be out there and they may not be so impossible. And so some of these things have come together in the last decade or so. And Dr. Urshik's discussion was a great example of how he's looking at developing these digital resources across the tree of life for some of the most extreme cases of body size and body shape evolution across the animal kingdom. Think about that for botanical studies. What would that actually take? I think the I don't know. I want to. There's part of me that wants to return the question to you and challenge you to think about exactly what the real what limitations are really there that could hold you back from accomplishing those types of goals. And thinking Philippines wide. But great question.
Thank you so
Thank you so
much.
much.
Hello, my name is Kenneth James Squirrel from University. I have a question. Does it you call it a new type of species or a genetically breed species? If it's, for example, like the popularity of breeding of snake breeds, like different kind of rainbow snake or articulated python. Does it count as a new or does it count as a new species or does it or is it like a genetic mutation
Okay, you're talking sorry, the
from Dr. Brown?
herpetocultural crowd of new snake coloration varieties into the Manila herpetocultural community, right? I think of those as new genetic variants. The interesting thing about them is that a lot of them come from natural variants. And so, no, I don't think a ball python that is all white and has black saddles on it is a new species compared to a wild type ball python. But the interesting thing is that somebody noticed a color pattern variation from a wild population, caught that snake, and bred it in captivity to produce that color variant that is now selling for an outrageous amount of money in the pet in the pet market. Um, so I think that that's an opportunity for you to think, no, it's not a new species. It's just a color variant. Do I really want to spend that much money for a pet snake?
Yeah, yeah. Who would spend $100,000 for a rainbow snake? Another
Great.
question from Dr. Jan. This is from this is from another question. How do we digitalize some extinct animals like Tibetan or Tibetan or um, Tibetan wool, a dodo bird, for example, like 100 years. How do we construct and digitalize it to make it more, especially if there's a term of coloration or body structures or inside? How do we do that? Just as a
So
question.
that's a great question. I don't have all the answers. We know that people in the past have done reconstructions of extinct animals. I think one big difference that we're trying to do is we're really trying to make sure the work is open access, and also put out in a form, in a format that people can use. So we actually have a paper we're working on with one of my grad students where we did do a reconstruction of the ivory-billed wood, or not, the woodpecker that was extinct in Mexico in the 1960s. And we did that based off a physical specimen, but also video of that animal in 1956 and other data. And so obviously, you know, it's a reconstruction, but we think it's a pretty accurate one. And that is available online for other people to use. And as you say, there's all kinds of levels you can do. You can add bone. We actually have a project with turtles where we're adding in muscle from CT scans and bones to models. So I think there's just different levels of hierarchy and structure you can do. I think it's very exciting to combine different kinds of data like CT scans with photogrammetry, with MRI, with photogrammetry. And I think it would be really interesting to apply those techniques to dinosaurs, and to create open access, high quality 3D models of dinosaurs that people could access and use to study and research. The only reason I haven't done it is I just don't have any time. So I think that with all the work in paleontology, that would be really interesting. I'm actually not aware of any open access, free, scientifically super sound 3D models of dinosaurs. Maybe I'm mistaken. I would also argue that the one thing about 3D modeling is it has to be done at a certain level of resolution and scale and consistency. So you can download 3D models of all kinds of animals online, but the one thing you'll quickly notice is they don't really have the same, there has to be a certain level of scientific accuracy and consistency and resolution. And I think that's really important too. So I think there actually are some studies and there's some library books and reviews coming out that are setting for some standards for 3D data. And I think those are going to be really important standards to establish so that we can have a common language for 3D modeling. And I think there's also going to be more common usage of certain 3D modeling formats, which I think will be really exciting. I know the FBX model came out a few years ago, but there's still not really one common standard of 3D modeling like there is like a PDF or a Word doc. I think that's coming. I think you're going to see that in the next 5 or 6 years too. So in short, it's a great question. I think extinct animals would be amazing. I think it's a great area for further research and development.
Quite sad, actually. Oh yeah, one final question. Might be a little bit sensitive to you guys. Is it possible? Is it gonna be considered as a new species, considering the failed project of reviving back the northern white rhinos? Same like the news. Just saw, as I see a picture, I just saw like a lonely male rhino, just a female died. Will it be no, like as a just actually be actually happening for that, but actually failed? If there was a possibility that a northern white rhino and like some kind of another different kind of type of rhino, like African white rhino, possible to breed a female or male, possible to breed, will it consider be a new type of species or will it be a separation of DNA? To Dr. Brown. Yes, I know. I was actually thinking of that while thinking of rare species like Visayan spotted deer. I was thinking like, will it be possible to cross genetics, crossly genetic from any type of deer? So will it be considered as a possible of a new type of genetics, or will it be a possible of like a different kind of breed but same kind of parents?
I know these kinds of questions are really are hard to what should I say, hard to resist for conservation, particular conservation community, subsets of conservation community. What do we do with things that we may revive from extinction? And there's all this interest in whether woolly mammoths could be, their DNA could be all sorts of other things, about other ways to think about reviving some footprint of the genomic variation from extinct species. I think the answer is no. It's not a new species, and we shouldn't think of it as a new species. We should think of it as reviving some aspect of the genetic evolutionary history of past extinct species. And so, yeah, I think we need to keep our ambition, or our sort of what am I trying to think? We need to sort of keep keep a reality check on what this biologically really means or what it represents. And if it's not a natural product of natural selection and millions of years of evolution, we shouldn't think of it as a new species, right? It's something that was bred in captivity under abnormal conditions and likely in suboptimal conditions of something that is not viable biologically anymore, right? Does that make sense? I don't know if that answers your question,
because
but I don't.
yeah, because
We shouldn't
I was thinking
confuse
for that.
the question of new species versus biologically partially resurrected genomic backgrounds of extinct species. I don't think we should consider those. We shouldn't conflate those issues.
Because I was actually thinking for that from Thailecan, because there's some mix of Thailecans, like even though they are officially extinct, so I was thinking like maybe a DNA of a Thailecan from a different kind of same genetic line as some lift of DNA. So I was thinking if a possibility that a DNA of a white rhino was supposed to be inserted from an African rhino. So there's like a slightly like chance of genetic, or in the future it might be like a clone. So we might have possible of like a revival of white rhino, or even close to extinction, spotted white, Visayan spotted.
All right, thank you so much. Seems that we don't have enough time to address your concerns and questions. Well, thank you so much for the rich exchange. The question and answers we have heard today reflect the depth of this community curiosity and passion. Okay? And now we are going to proceed with the awarding of certificates to Dr. Rafe Brown and Dr. Duncan Ershik. I'm going to read the content. Father Saturnino Arias University, Butuan City, Philippines. Certificate of Appreciation is awarded to Dr. Rafe M. Brown for delivering an engaging talk at the plenary session on biodiversity documentation and digitization during the first Philippine Biodiversity Data Congress, with the theme Field to Cloud, the Future to Open Access Conservation, given this 8th day of April 2026 at CBE Function Hall, Father Saturnino Arias University. Signed by our University President, Reverend Father Randy Jasper C. Ojik. Another certificate of appreciation is awarded to Dr. Duncan J. Ershik for delivering an engaging talk at the plenary session of biodiversity documentation and digitization during the first Philippine Biodiversity Data Congress, with the theme Field to Cloud, the Future to Open Access Conservation. Given this 8th day of April 2026, CBE Butuan, CBE Function Hall, Father Saturnino Arias University. Signed by the University President, Reverend Father Randy Jasper Theotek. And now, ladies and gentlemen, we will now take a 20 minutes break. Please help yourself to the refreshments prepared for you outside the hall. So we encourage you to connect with fellow participants, exchange contacts, and continue the conversation sparked by our first two speakers. We will resume at 11 with our next plenary session.
Tanakwa, Luzon, Mingan, Amuyao. So if you'll notice, these are all Luzon data. We don't have much data on Mindanao, and this is opportunity for you to start your work, for biodiversity in Mindanao. So we use all this data. So we use all these methods. So basically, we go out in the field, set trap, capture nets and everything, and then identify. That's similar to the way Dr. Brown has described how they collect data in the field. And this has given us more information and understanding somehow the ecology and where they are, and what are the type of habitat they need or food they need. However, these types of methods have its limitation. One, it's labor intensive. Those who have been doing field work, it's really, we stay, when we do field work sa mammals, we stay 6 weeks out in the field, walang uwian, covering the entire elevational gradient ng isang bundok. It's expensive, labor intensive, expensive, and very limited yung space mo. And we don't have temporal data. So yun yung very limitations, ano? And it's a challenge actually, considering that the rate of destruction, these conventional methods somehow are not able to cope up. A lot of questions that are being asked, like how do we measure our impact? And sometimes, or oftentimes, the answer is that we don't have much data to provide you or to calculate the indices or the metrics for us to respond to those kind of questions. And the government, the private sector needs that in order for them to, or us to develop the kind of actions that we need to protect our biodiversity. So basically, we don't have the luxury of time to, don't take me wrong for this, but taxonomy is actually very vital. But again, we need to respond to the needs, the changes, the fast changes of the climate, yung ating habitat destruction, deforestation. We need to cope up with those. And just to give you an idea, so this is a graph of showing rate of extinction. So yung dotted line yan, that's the natural rate of extinction, based on the 5 extinction rates that was recorded previously in the past in the millions of years. And right now, this is the rate of extinction that based on the calculation of IUCN. So it's more than 100 or 1,000 times double. We need to cope up with those threats. So that's where technology comes in. So now, these technologies are already actually available. We just need to really train ourselves, tap it, use it, for us to give a different perspective, different insights. Because it's important that, okay, we know the species, they're there, but the next question is how do they respond to those changes? In order for us to conserve or protect the species, we need to know how those changes impact those species. Ano ang dapat nating gawin to prevent the extinction? So this technology fills in the gap ng conventional natin na mga the basic or conventional data also gives us the insights about the species habitat or behavior, but it's a bit slower. And technology, the available technology right now, somehow gives us an edge. Like camera trap. For most mammals, they are nocturnal. And we work 8 to 5 in the morning. Yun lang yung time natin. Beyond that, overtime na. Hindi tayo pinapayagan pag nasa gobyerno. So sometimes it's difficult, particularly for nocturnal animals. And camera trapping, presence and absence would be a great tool. You can also actually not just presence of absence, but also monitor health of animals. There have been studies of using camera traps, and tracking yung mga ano no, diseases and injuries ng wildlife. Acoustics. So bioacoustics. So there are a lot of studies looking at the interaction between different species by just using bird calls. So each bird naman may unique signature na acoustic siya. So we can record that. Then you just leave the equipment doon, then balikan nyo. It's less yung labor work. And you can combine this and looking at this particular study, they look at ano yung soundscape. Ano, so yung sound ng insects, birds, mammals, and different habitat types or different land uses. And they have different responses. And it's a good, you don't need to really have a detailed species ID. You just look at the natural sound and unnatural sound. Then you can see how different land uses impact biodiversity. So so nalilesen natin yung dependence on just by kailangan identify. But if you want to go through in-depth study for a particular species, then you can go. Kasi nga, you can identify naman the particular bird or the particular mammal that you want to study. But this is a good tool for enhancing our capacity to monitor. So there's one student from an undergrad student. He did during the pandemic. Nabutan siya ng pandemic. So one of our partners from the National Institute of Physics, they come up with their DIY sound recorder. You can buy this Raspberry Pi. You can buy it sa Lazada, Shopee. Itong microphone na yan, di ba? Usually ginagamit niyan, nasa Lazada rin. So it's a matter of doing DIY. Then wala siya. So we just left the units sa different parts sa UP Diliman, then recorded all the data. Then after pandemic, nag-gather niya yung data, then computer-based na siya, then do the analysis. And it helps kasi you can identify yung what they call anthropony. These are human sounds, biophony, these are the nature sounds. So you can identify, and then they have different indices, that just like the species index indices na ginagamit natin. You can also do that for soundscape ecology. So we also compare that. We have a station in Palanan. It's an old growth forest, and compared it with the urban setting. So you notice the difference between the biophony, where there's a lot of natural sounds from birds, frogs, and probably bats. Then compared to construction work, so you can compare and you can see how. And then if you have longer data, you can have a dry season, wet season. Then you can see changes through time, and it helps understand how species respond to various anthropogenic activities. And you can combine these two, camera traps and acoustic sounds, and it gives you a more different or bigger picture. So the one yung hindi nakikita ng camera can be recorded by the sound. And if you combine that, then you have a better understanding and the dynamics of your ecosystem or your land use. Ang galing ng AI, sabi ko lang kailangan ko ng gumawa siya. So this is the conventional way when you do surveys. This is what you see. And with the technology, there are more. Parang ganun yung sabi ko, gawan mo nga ako ng illustration, na graphic to convey this message. So ginawa niya ako. So the succeeding slides would be more on the things that we do now, na mga technology that we implement to gather biodiversity data. So one of the instrument that we use is the use of telemetry. So we have this radio telemetry. We tag the animal and then track them. but for flying foxes, these are fruit bats. They are nocturnal and they travel along very large distances or large area, move a large area. So there's a telemetry that has a GPS tag, which I think Dr. Ibanez would later explain. But basically, because they're nocturnal, so we need a system na we can program every night na every 10 or 30 minutes interval, magda-download siya ng coordinates habang siya ay nagpo-forage. That's why this is how we come up with all this. So yan yung mga pinuntahan nila. So yan yung mga areas. And then we went to that area, tinignan namin ano yung land use, ano yung mga potential food items. And of course, this is part of in partnership with UPLB when we do biosurveillance for potential spillover. So it also adds yung health aspect nung study or nung ating monitoring. So we're looking at different potential spillover. Actually, karamihan ng pinupuntahan nila are tourist area. And so there's high potential of spillover. And you can overlay yung saan ba yung mga pig farms, saan ba yung mga and it's a good policy din na later on the agricultural sector should have require yung mga bat-proof na mga facilities nila kasi nga doon natatransmit minsan yung mga viruses through the domestic animals and then to humans, so mga ganun. So there are implications that we can or policies that we can do. And another thesis that we're working on somehow extensively because I have a PhD advisee doing this right now. So recent studies indicate that we might have 3 species of tarsier in the Philippines, not just one. Based on currently on the genetics, the Zamboanga area, the Dinagat, northern parts ng Surigao, and then the mainland Mindanao. There might be so in order to kasi if you develop an action plan and you have this kind of data, we need to be specific. So when we do conservation, we need to understand na baka hindi siya yung actions natin sa isang lugar, hindi siya applicable sa isang lugar. Because there's a potential na it's a different species. So this is what we're doing right now. And most of the work, so we have started working on tarsiers, basically tagging them. So ibalik ko lang. Hindi ko pala napakita. So this is the transmitter, the radio, VHF. And then this is a GPS transmitter. So dalawa ang ginagamit namin, mainly to track the animal. And second, since gabi siya, so there's a GPS logger, we call it na transmitter, that downloads locality every now and then. So may mga ganun So ang advantage nito, we can recapture the animal and remove the tag later on. Kasi may battery, lang yan 6 months. Actually, itong battery life niyan, 6 months. So before 6 months, we recapture the individual, remove the transmitter, then let the animal again move on its way. So these are the studies that we have. So we have identified their home range, interaction between male and female, and it seems they're not territorial, but they have high fidelity sa sleeping sites nila. This one is in Mount Matutum, yung Tarsier Sanctuary sa Tupi. So you may be familiar with that one. So these are the sleeping sites. And dyan lang namin sila na-observe. Hindi sila lumalayo dun sa sleeping sites nila. So these are based on sa telemetry kung saan sila yung natutulog. So the following day, pagtag, the following day, hanapin namin sila, nandito ka ulit. So we were able to. And it has an implication in monitoring. A lot of our monitoring, pag dating sa tarsier, sabihin, ay wala sila doon. O kaya, dahil hindi nila na-encounter for several months or several years, ay nag-decline yung population. And one way to monitor Philippine tarsier is to identify their sleeping site. Because they have high fidelity ng sleeping sites nila. So when you do monitoring, you just keep on going back to doon yung mga sleeping sites nila. You don't go there randomly. So those are some of the applications of such studies. And here, ang classification kasi ng tarsier is near threatened based on IUCN, but based on the Philippine Red List, sa National Red List natin, it's least concern. Here, what we're showing, so yung white na yan, our topography, although hindi sila ganun sensitive sa disturbance, but they still require vegetation. Kasi these are gali. So usually, di ba yung sa forest fragment natin ang natitira, yung mga gali, nandun yung vegetation. And their sleeping sites is doon sila connected. So they still need vegetation despite of they are quite tawag ito, hindi sila sensitive sa disturbance. They still require vegetation for them to survive. And this one, which was recently published, sa Siargao naman sa mangrove. So tarsiers can be also found in mangrove areas. And so similar naman yung attitude nila. They have high fidelity sa sleeping sites. But the food items, when we did a fecal analysis, because we have samples gathered, ang interesting is that dominant yung crablet. So sa forested area, they eat insects, other mammals, small mammals or lizards. Here, pagdating sa mangrove, crablet. And this data, don't have to explain it in detail, but basically it gave us such opportunity since we were able to tag a female with an offspring. So yung kanilang behavior, we were able to observe yung activity ng mga nung pair. And we are also have the opportunity to gather yung acoustic. Kasi when you identify sa taxonomy, right now, aside from the morphology, you have the genetics and you have the calls, even for amphibians. So we have to combine those. So there are some slight differences, similar shape. This one is from Bohol, this one is from the Initaw. Yung isa sa Bohol yun na recording, na published data, this is from Initaw. So there are some slight differences. We don't have enough data yet, so we're still gathering all this data. So that brings us to bioacoustic. So right now, our lab is actually building a bat library call. Similar sa birds. Di ba sa birds sa Sinocanto, pag pumunta kayo ng website, yung sa Cornell University na you can download yung mga calls ng birds. Yun ang lacking sa Philippines. In European countries, they have this automated, maglakad ka lang sa gabi, pag na-detect niyo yung call, sasabihin niya, eto yung species. And that's the goal for this one. We have been starting doing this. The challenge is, there's always a challenge. Similar species, Arquatus, different calls. So it's complicated. Rhinolopus bacrotis, very similar in morphology, external, but different calls. So those are challenges, And worse, different islands have different calls. That's why Mindanao, it's an opportunity for you to get into this kind of, those are some of the challenges, So from there, we also do, so this is an example, these two species, kung titingnan nyo sa literature, they are rare. We're able to record their calls. And then when we went out other site, nag-record kami, they are quite common. We have a lot. Sa Negros, they're quite common in Negros. I think in Leyte, very common din. But these species actually kasi, kaya less siya nakaka-capture kasi they are canopy dwellers. So if you don't do canopy nets, chances are you don't record this in the site. And that's the advantage of having that acoustic. Mas nare-record nyo yung mga hindi nakaka-capture ng actual. And yeah, so hindi lang tayo nagsistay dun sa ano. The reason why we wanted to do that, a library of calls, because later on, the next step is that you can now use it to monitor population. This study from one of our undergrad, this is an undergrad thesis, was able to, based on the calls, because we were able to identify yung calls, yung bat based on their calls, she was able to identify the abundance per species on a particular cave. And it's a good baseline because this is Biak na Bato National Park is being visited because of the cave. So it's important that they will have a baseline on the population density or abundance ng mga different species sa different caves nila. So we have already somehow a baseline for Biak na Bato. And the next step, we are now currently trying to automate. So another researcher in our lab has done programming, medyo savvy sila. So they did a script in R to help automate identification. So may script na yan sa R, just plug in yung data mo, range of acoustic, then he can draft anong species siya. So it's a step towards that goal of having our own automated. And later on, the idea is that it will be uploaded to a central database that can be applicable, that can be accessed by the public. So sa Australia, meron silang mga ganun, yung Kairobook, where you can actually access yung different data on acoustic calls. So right now we have about 38 out of the 54 species, but mostly sa Luzon lang. So there are still a lot, but at least we have this, and you can start comparing it with what you will have in the future. Camera traps. So we have also did some study. Ito naman, no, naging curious lang yung conclusion nitong paper, naging curious lang yung tarsier. Hindi not necessarily nakita niyo yung infrared. Kasi ang question kasi is, do camera traps elicit yung change in behavior sa species? So may mga observations na somehow curious sila, tinitigan nila. Ano ba itong bagong structure na nasa environment namin? So we also do camera traps basically for Dinagat. We have a project in Dinagat for 3 years. So we wanted to estimate population density ng different small mammals. And these are, I don't know, camera traps, snapshot. And alam niyo to, ano to? It's a feral cat kasama ng mga endemic na daga. Notice this space here and notice this area here and ito. So he is also using the trail that's being used by. So if you don't have the camera traps, hindi natin nalaman, uy, may feral pala yan. It's a threat to biodiversity. It's a, kasi yan yung major. So kaya nga healthy, no, makita namin masyado siyang healthy. And then we also use camera traps to see ano ba yung potential dispersers ng Rafflesia. So this is Rafflesia consueloe sa Pantabangan. So what are the potential seed dispersers ng Rafflesia? So we also use camera traps. And in Leyte, we're also doing population density estimates of warty pig. So those are things that we are currently doing. We still have time ba? Ito lang siguro yung tupaya. Yung sa pinakataas. You can click that. May cannot download information you requested. Sorry. We just moved to the next, siguro. We just moved to the next. So hopefully. So, ano ba ang common ulit doon? Aside from nocturnal sila, it's still somehow labor intensive, but less than the conventional. You still go out, you still set up the camera, then babalik ulit kayo, and then process the data. So it's somehow still, so we come up with this idea of coming up with a multi-sensor. So packing all this in one sensor. So we have, so the idea is that for us to be able to understand yung soundscape, combine it with human activities and natural sounds, we can be able to understand yung the dynamics ng landscape natin and the climate within the landscape. So ito yung idea, no? So sorry, ba't nagkaiba yung na-transfer kasi. But anyway, idea is camera trap, conventional, and data, coming up with an edge computing. Yun ang sabi natin. So basically, in a layman's term, parang ang gusto namin gawin, mag-deploy ka ng sensor doon, uuwi ka sa office mo, mag-aantay ka ng data. That's the idea. That's where we wanted to do. So we have a prototype, very crude. Ang challenge lang nito is the power source. So we have the Raspberry Pi, which serves as your computer. And you have this unit na available din sa Lazada as your microphone. And we incorporated temperature and humidity. And you will be able, then, once na ma-record niya, ibabato niya sa Google Drive mo. Ito yung recording, every 10 minutes na yung nagda-download. And then, may kaakibat siya na temperature, environmental data. So these are examples. So sa matutom ito, pero hindi yata mag-play. Mag-play kaya yan. Ayan. So passerines, dahil umaga yan, may manok. Gabi siya, though, that's a scaps owl. Call yan ng scaps owl. If you can play, siguro, kung kaya. So, nare-record siya. So, yan yung mga binabato na data sa computer mo. So, upo ka na lang sa office, mag-antay ka. So, we have several versions kasi ang question is, paano natin i-waterproof? Although, the next step is, ang ano namin is 3D printing. And depending kung ano yung gusto mong ilalaman dyan, so, the size would ano. So, hopefully, kaya kayo ma-access. So, we have 2 units sa Davao, sa Philippine Eagle Foundation. Sige, i-click mo nga. Oh, ayaw niya mag-work. Ayaw, hindi kaya. So anyway, ano lang sa ipapakita ko, kasi ano ito 11:38. We have data 11:30. Ngayon. So every day yan, since 2025, running yan hanggang ngayon. So it's a promising so it's a proof of concept na it's working. It works, It helps us, So we have also one in UPD. So again, ang challenge nga is yung source of source of power. Yun ang pinaka-challenge. So we are also trying to come up ano ba yung power na ginagamit niya. Then we'll see if solar power will be doable or kaya niya i-manage yung power consumption. So last few slides. Of course, with greater data comes greater responsibility and greater. So familiar, Spider-Man. Mahilig ako sa Marvel, mahilig ako sa mga anime. So again, ang sinasabi lang nitong dalawang papers na to, we have to be consistent. We have to make sure, kasi nga ang daming technology. We need to have a standard. Kasi if different technology, you use different technology, yes, you will be able to get it, but you cannot compare those things. If different technology ang gagamitin nyo, different metrics ang gagamitin nyo, how do we compare? And we need a long-term data that tells us patterns. how they respond to these changes. And there are, it's a good read. If you, it's available online and it's free. You can, it's an open access. And they have this this, ano, no, yung parang framework to follow. So for the summary, we still need baseline data. Again, absence of evidence could be interpreted as evidence of absence. So baseline data is still very much important. Okay? So hindi natin nira-write off yung basic biodiversity surveys natin. We still need those things. Consistent long-term monitoring, sustained. Kasi minsan ang nangyayari, project-based, beyond project-based, wala na. And then wala na. So we need to think, when we do our proposals, we need to think longer term. You can do short-term projects, but you have to have a longer plan for you to sustain the kind of research that we need to do to help conserve our biodiversity. And of course, yung tech is not a replacement for biology. Diba? It's only a force multiplier. So we still need young conservationists, young biologists. Puro kayo computer, sabi kami medyo, yung ano namin, mga 128 MB na lang yata kaya namin. Sa inyo, malaki-laki pa yung capacity nyo to. Ano. No, we have done a lot of work and we still need a lot of things to do in order to conserve our biodiversity. Because in the future, collecting more data is just not enough. We need better standardized and ethically managed data and convert this data to policy. Dapat yun ang end goal eh. Yung survey mo, hindi lang mag-end sa manuscript, sa publication. It should have an application in terms of policy. Yun dapat ang nating iniisip. And lastly, these are the people I work with. Yung iba nag-gain na ng weight, yung iba hindi naman. So these are the people I work with through the years. These are members of the Biodiversity Club. Some of them have went to study abroad. Some of them got married. Some of them kung saan-saan. And ito yung pinaka-latest na group that I'm working with. This is 2025. So, yeah, I think that's all. And I'm happy to answer your questions. Thank you very much.
Dr. Doya, thank you for a truly illuminating presentation. And now, since we're running out of time, we will entertain 2 questions only. Okay. Dr. Doya, please stay sa podium po. Yan. So, who would like to ask question? Dalawa lang po ang entertain. Sige, please. Proceed to the microphone at the center.
Yeah, good morning, Dr. Duya. Yes, I'm A.Z. Makaling from the Ateneo de Davao University. So these are my questions. Actually, I have 2 questions in Connect. Can we use the overall complexity of the soundscape as a proxy for ecosystem health? For instance, does a quieter or less diverse soundscape correlate directly with a loss of functional diversity in that specific habitat?
The answer is yes. Yeah, because you remember there are different indices for the landscape soundscape. There are different indices, and you can use that indices as proxy to determine the health of the ecosystem. So basically, the more natural sounds, the more healthy the environment. The more anthropogenic sounds, the less. So may mga ganong pwede mong i-drive at with all those data without really just looking at specific species, but the diversity of natural sounds can tell you the status or the state of the environment where that area is.
Thank you so much. And in Connect for that, questions. How well do, like we are talking about indices, like the acost or like those acoustic indices or bioacoustic index actually correlate with the traditional physical surveys? Are these cases where a soundscape sounds healthy but the actual species richness is declining?
I don't know if there are still comparisons, but again, so soundscape when as I've mentioned, if you have more natural sounds, you have more healthier environment compared to those areas that have more anthropogenic sounds. So if you compare it, that natural sounds, you need to go on deeper. You need to see what are the species producing that sound. So now you'll get the idea of, are these species widespread or are these species endemic to the country? Then you can further assess that somehow this is a secondary forest and this is a primary forest. So those are things that can be done using those indices and sound acoustic data.
Thank you so much, sir.
Okay, one last question. All right.
Good morning again. I am Shiri from Ateneo de Davao University. So, sir, how does pilot testing is very important, no? How does it improve accuracy and reliability in biodiversity research? And should it be a thing before we really get the results in implementing those technology-driven methodologies? Yung pilot testing po ng methods.
Yeah, as Dr. Rafe Brown mentioned also, that there is an ethical guidelines that we need to, despite that it's just a pilot, but you still need those basic like permit. You have to have an endorsement that community where you are going to work with. You have to have a discussion with them. I mean, letting them know that you're doing this one. So yeah, there's a basic work that needs to be done before you do all these pilot testing activities.
Another one last question. How do you balance in traditional field work and using technology-driven methodologies so that we don't lose the real context po in knowing the situation in nature?
Well, as I mentioned, basic science, taxonomy, basic surveys are still very important because you cannot identify that particular sound if you don't have that basic data. So you have to still have that basic data that although you can use sonotize, but then again, it would be more meaningful. It would be more species specific. Your actions would be more accurate if you know the particular species that's creating that sound. Okay. So there's still, hindi yun na ito na lang gawin mo or ito na lang gawin ko. It's more on, it's combining the two is the best approach.
Thank you, sir.
Okay, thank you so much. By the way, Dr. Doya, is it possible that because some of our participants would like to raise question or address their concern, can they email you
Yeah,
regarding
sure.
with their? Okay, you just ask the email of Dr. Doya later.
Or,
Right?
I'll be here the whole day naman.
Sige po. All right, so moving on po, we would like to invite, please, Dr. Doya, stay on the podium. We would like to request Dr. Marites Bunachita Sanggela to assist or to give the certificate. And okay, here's the certificate. Father Saturnino Orius University, Butuan City, Philippines. Certificate of Appreciation is awarded to Dr. Mariano Roy M. Duya for delivering an engaging talk at the plenary session on biodiversity documentation and digitization during the first Philippine Biodiversity Data Congress, with the theme Field to Cloud, the future of open access conservation. Given this 8th day of April 2026, CB Function Hall, Father Saturnino Orius University. Signed by the University President, Reverend Father Randy Jasper C. Ochek. Thank you so much, Dr. Doya. Your insights and your presence today have enriched this Congress immeasurably. Thank you once again. We have the pleasure of receiving a very special message of support for this Congress. This message underscores the importance of what we are doing here today and the institutional backing behind it. To give us the message of support is the Chief of Emerging Technology Development Division of the Department of Science and Technology, Philippine Council for Industry, Energy, and Emerging Technology Research and Development, or DOST PSHIRD. In this capacity, she oversees programs that accelerate the development and adoption of emerging technologies in support of national development goals, including those that directly touch on data science, digital tools, and research infrastructure so vital to the work that we are presenting at this Congress. Ladies and gentlemen, the Chief of the Emerging Technology Development Division of DOST shared Ms. Edna C. Nascenseno for her video message.
Teachers, students, and participants, warm greetings. I am pleased to join you today for the Philippine Biodiversity Data Congress 2026, an important gathering that brings us together around a shared goal, strengthening biodiversity data in the Philippines through better documentation, digitization, and access. This Congress comes at a crucial time as biodiversity loss continues to challenge us. The need for accurate, accessible, and well-managed data becomes even more urgent. Biodiversity data underpins effective conservation, supports scientific research and education, and enables sound, evidence-based policymaking. At Bashir, we recognize that addressing environmental challenges requires the integration of science, technology, and innovation. Over the years, we have worked with partners to support research initiatives that apply emerging technologies to national priorities, including biodiversity conservation and sustainability. One key area we continue to advance is biodiversity data digitization. Technologies such as macro photography and photogrammetry are transforming how we document biological specimens, allowing for high-resolution imaging, detailed morphological analysis, and even three-dimensional representations. These innovations enable biodiversity data to be preserved, analyzed, and shared more effectively. More importantly, they help make our rich biodiversity more visible and accessible, supporting researchers, educators, and institutions across the country. This Congress reflects a collective effort toward that shared direction. In this context, we acknowledge the PSHIR GIA-funded project, Mabulig, or Mobile Augmented Reality on Biodiversity Utilization using digital lens for species identification, as one of the co-organizers of this event. Through its training workshops, Mabulig contributes by building practical skills in biodiversity documentation and digital imaging. These efforts highlight a key point. While technology is important, people are at a center. Strengthening biodiversity data systems requires developing the capabilities of students, educators, and researchers, and fostering collaboration across disciplines and sectors. And this is exactly what this Congress enables, a space for learning, exchange, and partnership building that can guide future efforts in biodiversity data. As we move forward, let us continue working together to harness digital technologies responsibly, expand access to biodiversity information, and protect our natural heritage. Lastly, I'd like to commend the organizers, speakers, and participants for making this Congress possible. and I encourage everyone to actively engage and maximize the learning opportunities. Maraming salamat, and I wish you all a meaningful and productive Congress. Isang magandang agham na umaga po sa inyong lahat.
Ms. Teshensenko, thank you so much for that meaningful message of support. And now, ladies and gentlemen, it is time for lunch. Lunch will be served outside. Please proceed in an orderly manner and take this opportunity to rest, reconnect, and recharge. Okay? We will resume at 1 o'clock in this afternoon for the session 2, where we will shift our focus to conservation and hear some truly remarkable leaders in the field. And to our online participants who have been with us through the morning, thank you for your patience and engagement. We will see you back online at 1 p.m. Enjoy your lunch, everyone. Good afternoon, everyone. So welcome back to the Philippine Biodiversity Data Congress 2026. Please settle down. All right. By the way, we have the workshop simultaneously happening right now at lobby. We have the field experiential learning, a simulation of Philippine biodiversity documentation. At the lobby, we also have the biodiversity booth. For the digital imaging photogrammetry and 3D modeling, dito po sila ngayon, bumababa na po from the second floor. Welcome back. In a couple of minutes, we are going to have the afternoon session for the plenary session too. All right. By the way, guys, should you need to use the comfort room, it is located at the back of this hall. Yung I hope that your lunch was nourishing as this morning's discussion, and we have equally rich afternoon ahead of us. Our afternoon session for the plenary session 2 turns our attention to conservation, the ultimate purpose behind all the documentation, digitization, and data work we have been exploring. Because at the end of the day, every data point, every 3D model, every GPS coordinate exists for one reason, to help us protect life on this earth. Okay? Our first speaker for this afternoon needs very little introduction among those of us in Philippine biodiversity circles, but he certainly deserves every word of it. He's director of operation of the Philippines Eagle Foundation, the organization at the vanguard of protecting what is perhaps the most iconic symbol of Philippine biodiversity. The Philippine eagle, our national bird, and one of the most powerful endangered raptors on the planet. For years, he has worked tirelessly to combine rigorous science with on the ground conservation action, studying the ecology of flagship species, navigating the complexities of protected area management, and bridging the world of research with the realities of conservation in developing countries. His talk, his talk on the impact of technology on the conservation of iconic flagship species will show how the tools of modern science are being deployed in the most critical conservation battles. Please welcome on the stage the Director of Operation of the Philippine Eagle Foundation, Dr. Jason C. Ibanez. Sir?
Okay. Thank you, sir, for the very generous introduction. Magandang hapon po sa ating lahat. And again, thank you to Father Orius University for the chance to share our story. And part of my story is to argue that I guess the greatest threat of humanity, of the human species, is not actually speed, strength, or size, but it's our ability to do innovation. And we personally believe that at the Philippine Eagle Foundation, that innovation can really make a difference in the survival of our dear icon. So my talk is about how the Philippine Eagle Conservation Program has benefited in the latest years on the use of technology to really look at the conservation needs of the species while also creating local government, national government, and Filipinos and international collaborators constituency for its conservation. So my presentation argues that the Philippine Eagle is a very practical example of how technology and modern scientific tools can really make a difference in the status of an imperial species. No, we all know the Philippine eagle. It's one of the largest, and I can say most beautiful raptor in the world. Of course, I'm biased. It's as Filipino as we are all Filipinos. We don't have tigers. No, we don't have the charismatic tigers, the big lions of Africa, but we have a bird of prey. No, as our apex predator, as our top carnivore, and we all know the value of the top carnivores in maintaining the health of our ecosystem, forests specifically, because I'm sure everyone knows that the ancient vegetation of the Philippines is essentially forest. We're a forest archipelago, and that's why many of our endemic and endangered species, including the Philippine eagle, is very much forest dependent. And in order to save an icon, a keystone species like the Philippine eagle, we need to save the forest and large chunks of it. So, from the system's perspective, ecosystem or social ecological systems perspective, using the Philippine eagle as a gives important insight on how indeed technology and tools can help conserve species. And what I would be sharing would be almost 4 decades of experience of the Philippine Eagle Foundation, showing that technology can really help save species. My story is also about showing how in each aspect of the conservation work, technology has contributed. So for a rare species like the Philippine eagle, finding each territory or nesting territory is very critical. Why? Because Philippine eagles are very loyal to the places where they breed. They use the same nesting site over and over again. And that's why it's very important for reproduction, very important for securing an area where Philippine eagles can breed safely. In the same way that we want to be protected within our own households, within our own backyard. The Philippine eagle, each monogamous pair of a Philippine eagle needs a safe breeding or nesting area so that it can perpetuate its species into the future. And technology, as you would see later, is helping us find and set aside these species. Now, we also need to track where they go. Tracking allows us to identify which parts of the forest are important for the species. You know, how they use the habitat, their habitat use patterns, how big an area they require. And again, in this anthropogenic world, no? In the age of Anthropocene, it's really very important to find yung sweet spot, the right balance between securing yung immediate needs, critical needs of the species, while also securing human needs. Technology that also helps us see population trends through time. That's very important for monitoring the success of our conservation project. Are we really helping the species? Is our conservation work really resulting to positive impact to the species? Technology must also allow us to identify threats. You know, according to the former president of the Peregrine Fund, the late Dr. Burnham, one of the minimum requirements for saving an imperiled species is first identify numbers and what are the threats that are causing it decline. And I would show how technology has helped us identify threats to the species. And also technology contributing to decision. Now, I had some conversations with colleagues in the crowd, and there was an emphasis on transforming, very important ecological data into informed decisions by LGUs, the government, and decision makers in general, including our indigenous peoples who own and have decision-making power over their ancestral domain. So I would show how technology can also contribute to that. Equally important is community participation. The use of technology should also be inclusive and should also result to positive impact, including material benefits to communities, including indigenous communities who might be hosting our conservation work, and then repeat that process. Okay. So uh, the first tool or technology that really helped us a lot in recent years is the mapping, modeling where Philippine eagles exist. Our colleagues from the Peregrine Fund helped us do a species distribution modeling for the Philippine eagle. And finally, we now know which of the remaining forests are actually highly suitable eagle habitats existing. So this is a general flow of the species distribution modeling. To be honest, this is not my expertise. But what it shows is that using old location data nest location, as well as GPS telemetry data from 6 Philippine eagle adults. The model was able to identify habitat features that are important for the Philippine eagle. No? Habitat covariates that apparently the Philippine eagles are selecting as their favorite habitat. And then the model was able to predict where these habitat variables, these habitat conditions are across the Philippines. And because of that, we came up with a magic number for suitable habitat. Okay. Now we know that there's about 2.8 million hectares of suitable habitat distributed across the Philippines. And from this area of habitat estimated for Philippine eagles, and based on our data on the average home range size of the Philippine eagle, which is about 7,300 hectares, using telemetry data, we know that Mindanao appears to have most of the suitable habitat for Philippine eagles. Mindanao is a stronghold for Philippine eagles. About 233 pairs could be found, assuming that all suitable habitats are occupied. Luzon has the second most number of population, about 128 pairs, and about 31 pairs if all habitats in Leyte and Samar is occupied. And these light green polygons represent, again, the suitable habitat for Philippine eagle. And the argument is that if you would want to look for Philippine eagles and see them in the wild and look for their nest, you must focus your search in these particular areas. And that again, as you would see later, have helped us optimize our survey method, because in the past we relied solely on interviews as well as historical data. Now, species distribution modeling has allowed us to identify which places, which forests could most likely have the Philippine eagle pair, and again, this optimized our way of doing our survey. Our colleagues from the Peregrine Fund also did some statistical analysis and sampling studies, and they came up with a specific survey method that tries to look at those suitable habitats, create hexagons, overlay hexagons as focal survey sites, and within each hexagon, which is about 3,600 hectares, subdivided into 7 subcells, and see how best to do a survey that could lead to a detection. And based on this study, this sampling study, it shows that out of the 7 sub-survey sites, all you need to do is concentrate your surveys to 4 subcells that has the highest suitable habitat, and that would reduce your efforts by 40%. Okay, and again, that's the beauty of mathematics and statistics. And I'm very pleased to share that by employing this modified or improved survey methods, we were able to detect at least 2 new nesting sites in Mindanao, one in Mount Candalaga in Maragusan. This is the newly discovered ancient nest. An eaglet is already on that nest when the nest was discovered last year. We found the first active nest record in Surigao del Norte last December in Higakit. And the local government unit, I'm pleased to share that they've now declared the Philippine eagle as their municipal flagship species because of this initiative. And then we've also detected soaring adults in Cabuaya, in Mati, an old nesting site, for the first time in 10
years. And again, in Samar, we did the same survey, we did the same strategy, and for the first time, an adult Philippine eagle was seen successfully, and it really shows that the survey method is working as far as Philippine eagle surveys is concerned. So again, this is, I think, the beauty of using technology, using statistical probabilities for optimizing Philippine eagle survey. And this has saved us resources as well as time in doing the survey. One of the also exciting aspect of our work is putting GPS tags, GPS transmitters on Philippine eagles. Now, since we started the program in 2008, we were able to tag 38 eagles already. And this has been generating data that's feeding directly into conservation work. So for instance, this is Philippine eagle Kalabugao. She was rescued from the mountains of Manolo Fortich in Bukidnon in 2009. And then a year later, after successful rehabilitation, we installed a GPS transmitter as well as radio transmitter on Kalabugao. And 2 years later, we were able to document how she ended up finding her mate, and on top of that, also establishing their first active nest. No, so natulungan po natin si Kalabugao to find yung forever niya. So this is the GPS track of Philippine eagle Kalabugao. In 2010, after successful rehabilitation, nirelease po namin siya sa Mount Kitanglad together with the help of LGU Sumilaw, our host, and the PAMBI of Mount Kitanglad. After a few months, nag-decide pong mag-cross si Philippine eagle Kalabugao to the other mountain range, which is the Mount Tago mountain range of Manolo Fortich. And for 6 months, Philippine eagle Kalabugao actually used the riparian forest of Kulaman River as a dispersal pathway. And this is the first evidence that for juveniles or for immatures, maybe because they're still inexperienced, they cannot fly outright from one mountain to the other. They would need stepping stones or corridors in order to disperse successfully. And the fact that she used the riparian forest for 6 months means that the riparian forest was able to provide for its ecological needs. And I'm very pleased to share that now, in many of the forest land use plans and CLUPs that LGUs, our partner LGUs have been adopted, they have set aside riparian forest as a form of protection forest within their municipality or city. And riparian forest conservation is slowly becoming a popular way of ensuring connectivity between two isolated habitats. Again, telemetry also has helped us justify for a corridor approach in conserving important forests. In this example, this is Philippine eagle Malambugok that was rescued during the pandemic. And then after successful rehabilitation, we were able to release her back to where she was rescued in Caraga, Davao Oriental. Malambugok is an immature Philippine eagle. And again, as you can see in her dispersal pattern, she's actually using a stretch of forest corridor as her dispersal pathway. While adult Philippine eagles are very loyal to a certain area of territory, it is actually the immature birds. Now, once they become independent, they're the ones moving across the landscape and until they become sexually mature and find their own mate. And so from the perspective of forest conservation, if we really want to maintain connectivity as well as gene flow between mountains, it's really very important to protect forests and ensure that there's a corridor or a connectivity um, between these forests. And this has become an important aspect of land use planning among local governments and including the DNR. Okay, another way of using the species distribution model for Philippine eagle conservation is its value for predicting areas that are suitable as release sites for Philippine eagles. In this particular example, students from the Department of Engineering in UP Diliman used our data for species distribution modeling and other habitat variables that are important for selecting release sites. Unfortunately, out of the 4 islands where Philippine eagles are found, we think that the Philippine eagles have been lost already in Leyte, primarily because of the impacts of Typhoon Yolanda in 2013. Now, if you recall my species distribution map earlier, meron pang adequate habitat present within the island of Leyte. But unfortunately, because of a catastrophe, we believe that all of the surviving eagles in Leyte has been wiped out because of Super Typhoon Yolanda. And now what we're doing is bringing back the species to Leyte because we found out that there's still enough habitats there. There's still prey items. This is a study that has been led and done by Dr. Aloy Duya and our colleagues from UP Diliman during the time of the late Dr. Perry Ong. And indeed, the habitat found in Leyte is still suitable for Philippine eagles. Now, the key question would be, if you will be releasing eagles in Leyte, where do you release them? And by identifying or analyzing the slopes, the ravines, the location of residences, no, again another modeling work, road buffers as proxy for power line risk, and then helipad access, just in case we want to fly in, no, a bird for release. The students came up with this distribution model of suitable release site. We're currently doing the release site. We're currently doing the release in Barangay Cagbana in Burauen, which was found to be a suitable habitat for release. We've released 5 eagles already, and 3 out of the 5 eagles were its release were successful, meaning they're now fully independent in the wild. We've lost Philippine eagle uswag. I don't know if you have heard about yung news ng isang release eagle that accidentally fell into the sea, the Philippine eagle uswag. So that's one result, no? And then I'm sad to say that one of the eagles was actually shot by a farmer. But 3 out of 5, successful release is already a good number. Two of the eagles have settled in Barangay Cagbana. So apparently they've chosen the forest of Barangay Cagbana as their nesting territory. So hindi ka na pwedeng mag-release doon. Now based on this map, we've identified the forest of Javier as a potential release site next year. And then we'll be releasing eagles also within Barangay within Dagami or Haro or Ormoc in the future. Because based on our projections, we need to release at least 15 eagles in Leyte in order to establish a self-sustaining population. Now, we've released 5 eagles, so hopefully, wish us luck, we can release 10 more eagles in the future. But again, modeling allowed us to identify, based on the best information available, where to release Philippine eagles that would potentially result to success.
Okay. Again, you know, information from modeling is really very important, but equally important, if not more important, would be for this information to be translated into land use decisions for the benefit of biodiversity. And there's no better example of this than what happened in Apayao province, which it has been declared as the fourth UNESCO Biosphere Reserve of the Philippines. So what the data on Philippine eagles did is to provide guidance on how to do the zoning of the UNESCO Biosphere Reserve in Apayao. So this is Apayao province. The green areas are actually protection areas that were identified using a map of the highly suitable Philippine eagle habitat in the province. So they've used yung pong result ng species distribution modeling, those habitats that were identified by the model as high quality habitat, they've set it aside as a core zone of the biosphere reserve. Those areas that are relatively medium quality habitats, they've designated it as a buffer zone. And for areas that are non-Philippine eagle habitat, they've declared it as a transition zone. And these are the three major zones of a UNESCO Biosphere Reserve. So again, the beauty of the project of this initiative is that in the absence of massive data on biodiversity, they've used the needs, ecological needs of the Philippine eagle as a reference for zoning. And I'm very pleased to share that the local government units, especially the municipalities, are now in the process of modifying their forest land use plans so that the zones identified within the UNESCO Biosphere Reserve gets affirmed and harmonized with the town plan. Okay, another example of how modeling has helped guide planning for Philippine eagle. Our colleagues from the Conservation Specialist Group of the IUCN Species Survival Commission did a population viability analysis for Philippine eagles. A PVA looks at the extinction probability of a species based on available data. And based on their analysis, they found out that the most important threat or immediate threat to Philippine eagles is high mortality rates for subadults and then juveniles. Based on the current data on eagles that are getting trapped and killed, their model predicted that if there's a 25% increase in subadult mortalities against baseline mortality, the Philippine eagle could get extinct in the next 50 to 80 years, which simply means that if we don't do something about shooting and hunting of inexperienced and naive juveniles and subadults, we would lose our national bird, even though we still have enough habitat that can sustain them. And this is what has been carried in the latest release of the Philippine Eagle Species Action Plan. We really need to do something with shooting and trapping and killing Philippine eagles because it can hurt the population within our generation. A few more slides about how telemetry can really inform the way we monitor and ensure the survival of Philippine eagles. Now, for a critically endangered species like the Philippine eagle, every individual counts. Every individual that survives can have an impact on population growth so that whenever there's an opportunity to release rehabilitated bird, we do it. And in order to really ensure that that individual survives, we make it a point to install GPS trackers and radio telemetry equipment or the transmitters on the bird. Um, and so I mentioned earlier about the 5 birds that were released in Leyte. These were all rescued birds that were successfully rehabilitated and released in Leyte to establish the population there. And I'm very pleased to say that the latest release, Philippine Eagle Lacue, a male eagle, an adult male eagle, has successfully hunted in the wild 2 weeks ago. Ito po yung first documented success hunt ni Philippine Eagle Lacue, mga prey remains from a sailfin lizard that Philippine Eagle Lacue has hunted. He also successfully hunted a palm civet. Meron po kasing radio transmitter yung bird, and so our team, mainly composed of indigenous Mamanwa residents and Waray residents, they were able to detect Lacue feeding on a dead palm civet. And then 4 days later, ito na po yung naiwan doon sa pagkain niya. And this really confirmed yung hypothesis that Philippine eagles can actually store their food and then return to the same food several days to consume it. And yun nga, after 4 days, yan na po yung naiwan, yung hindquarter. This is Philippine Eagle Lacue. The antenna is composed of, or the transmitter is composed of a GPS tracker as well as a radio transmitter. And that's Lacue for hunting from the ground. He's a male eagle. May dalawang female eagles na nandun ngayon. We're excited to wait for the breeding season. Baka po mag-agawan yung girls. Sino ang mapupusuan ni Lacue. Okay, again, we're doing our conservation breeding program. You all know that. This is our latest hatchling, si Philippine Eagle Bayani. If you want to know his status or her status, we feel na female po siya, although hindi pa ko ginawa ng DNA sexing. You can check out our live streaming. Again, our live streaming is hosted by explore.org of the United States. And because of the live streaming project, we're getting more followers and also more donations. And I guess that's another aspect of technology, its ability to build public constituency and also bring in funding. The conservation breeding program is still a very important part of our work. There's evidence that, again, because of isolation and breeding between close relatives in isolated mountains, the genomic diversity of Philippine eagles in the wild are very low. We have evidences of inbreeding based on 2 studies done by the Philippine Genome Center of UP Mindanao and the Philippine Genome Center of UP Diliman. This is the clustering of genomic data from Philippine eagles rescued from the wild and bred in captivity. It appears that some of the birds that we've rescued from the wild are already inbred. For instance, the Philippine Eagle CJ po, together with Lyra Sinabadan and Uswag, they came from Mount Apo, but it appears that they're very much related. And based on the homozygosity data, Philippine Eagle CJ is a product of inbreeding. No? And this makes sense kasi with the isolation of the many mountain ranges in Mindanao, it appears that there's no gene flow between the mountains. And this is very important because it can then guide us in terms of pairing Philippine eagles in captivity. And indeed, no, conservation breeding is effective at maintaining genetic diversity. In this particular graph po of heterozygosity, this cluster of heterozygosity levels, relatively high heterozygosity levels, are from captive bred birds. While for eagles that were rescued from the wild, no, bred in the wild and then rescued, you have evidences of very low heterozygosity level. Now, what conservation breeding can do is you can manipulate breeding so that
you get the most out of breeding these birds and maintain genetic diversity. Okay, I've mentioned earlier that it's very important for uh, technology to benefit local communities as well. Our forest guarding program is a very fertile entry point for introducing new technologies and modern tools to our indigenous communities. We're now working with 14 indigenous groups protecting 12 Philippine Eagle territories, and they've been using the same tools and techniques. They helped us in monitoring the Philippine Eagles. They get fairly paid for the service that they provide, and they're also learning these tools and techniques and the theories of it, no, through experiential learning. We've also adopted smart mobile phone-based app for monitoring species. We're very happy to be partnering with UP Diliman in exploring itong bioacoustics and camera trapping for the work that they do. These are our forest guards using the mobile phone apps and doing patrols. No? In each of the forest guard program that we're supporting, um, they've identified at least 14 species of flagship or focal species. No? These species are both ecological keystones, but at the same time, cultural keystones. No? Species that are culturally important to them because that gives the extra benefit of buy-in. They want to know the status of the species that are culturally important to them. Okay. So I think what I've shown is a collection of initiatives where clearly technology has benefited conservation actions. And most of this, if not all, had found its way into policy and improving decisions on how to protect lands as well as forests. And I would like to reiterate um, a, a summary line for my presentation. So it's very important to know what problem we want to improve using that technology, who will use the technology, and then if the technology is really suitable. But above it all, we also believe that while technology is powerful, purpose and people make it matter. A satellite tag can show us where an eagle goes, but only people can decide why that matters. Thanks again for the opportunity to share our story and magandang hapon po sa inyo lahat.
Thank you. That was Dr. Jason C. Benyes. And now for our second speaker for this afternoon, join us virtually. And her contribution to Philippine biodiversity conservation spans decades of dedicated fieldwork, institutional leadership, and scientific excellence. She is the executive director of the Philippines Biodiversity Conservation Foundation, an organization that has been instrumental in supporting the long-term monitoring and protection of biodiversity across key ecosystems in the Philippines. Her career is study in the power of sustained, patient scientific inquiry. She understands better than most how long field data gathered year after year, seasons after season, in conditions that are often challenging, becomes the foundation upon which effective conservation policy is built. Her presentation on the significance of long-term field data in biodiversity conservation efforts in the Philippines will speak directly to our hearts about why we're all here. Ladies and gentlemen, please welcome the Executive Director of the Philippine Biodiversity Conservation Foundation, Ms. Lisa J. Pangutalan Marte.
Good afternoon. Kumusta po kayo? How I wish I am there in person. But let me make it up to you by asking the first question. If I send you to Tubataha tomorrow, what data would you collect? And more importantly, what data would actually change how the site is managed? This is the Tubataha Bird Islet, where you have concentrations of seabirds that are actually breeding there, to reaching up to 15 to 18,000. That's the concentration of birds that you can find there, sometimes even more. Now for the harder question. Which of that data would actually change how the site is managed? Because we are asking this since we don't just have a data problem. We actually also have a translation challenge. We collect a lot of data, some of it useful, but only a fraction actually informs decision. Today, I want to share how we can bridge the gap from field data collection to policy development to real conservation outcomes. In the field, we are often faced with real constraints. This is a classic scenario when we survey poorly known sites, remote areas with difficult terrain, and we are only given a very short time to spend in the field to collect that data. We also sometimes have to do trade offs between doing some logistics and data collection. When I was still a student, I also have to cook meals for my team in makeshift kitchen. We are also constrained to document everything. In fact, sometimes we just document anything that we find in the field. We gather as much data as we can while we still can. And in the end, we actually don't know or sometimes at loss what we are going to do with the data that we have collected. But here's the key question in conservation science. How important is the data we are collecting if it does not influence decisions? For example, we know that some bird families, like the doves and pigeons, the flycatchers, the owls, the hornbills, have high numbers of threatened species. But knowing that alone does not protect them. So what kind of data does? We start with the hornbills. Hornbills are among the most threatened bird groups in the Philippines. All of them are endemic. Many of them are restricted to one or a few group of islands in our country. They are also very ideal for monitoring as they are large and huge birds. They are very colorful and they have loud and distinctive calls that resonates in the forest. And because of these attributes, they are detectable across distances. But they also occur across islands and landscapes, not just single sites. In many cases, these landscapes are extensive and requires a lot of resources. As hornbills are often found in areas that are inaccessible, we ask the question, can we actually generate population data that is meaningful for management at stake? At landscape level of population monitoring, this can actually be augmented. It is possible with citizen science participation. What we did was conduct synchronized counts using citizen scientists. We have volunteers or citizen science assigned for each specific transects within the monitoring trails within elevational limits of the species. That's within the range of 300 to 1,200 meter elevation for the Visayan Tarictic hornbill. As the teams are already assigned in places, we do the simultaneous count at 6 o'clock in the morning across different protected areas and forest patches. What we found for the Visayan Tarikti Cornville was quite unexpected for us because we have more than doubled the known population estimate of the species for both Negros and Panay Islands. So our count was at around 3,107 individuals for Negros alone. Why does this matter? This was not just a better number. The data set that was obtained challenges the global population estimates. It also strengthened the basis for protected area monitoring systems. In fact, the three protected areas of Negros, you have the Northern Negros Natural Park, the Mount Canlaon Natural Park, and the Balinsasayo Twin Lakes Natural Park, all have adopted the synchronized biomonitoring using also hornbills as an indicator species. And thus, a monitoring system of the identification of disease species across forest patches also helped identify new and retained the key biodiversity areas that are already existing in the Philippines. Most importantly, these simple and repeatable methods can actually generate policy relevant data at a landscape level. This approach was later replicated in Mount Banahaw de Tayabas in Luzon. Here we have farmers as our citizen scientists who also help record extremely high densities of Luzon Tarikti cornbills within the Mount Banahaw in the area of Tayabas City. And after 10 years of continuous protection and monitoring, we finally recorded the first group of northern rufous hornbills, or what we locally known as Kalaw, in one of the monitoring stations. It was quite a celebration for everyone because we really have hoped to see it. And one day, we never thought that it would actually appear, but it did. Not one, but 3 individuals. I will now share one of our example that is also very close to me. I really feel strongly about this because I've been involved for a long time with the black shama conservation. The black shama is an endemic bird of Cebu. It is a small black bird that has a melodious song, and its melodious song has earned it as a name of being a siloy. Siloy is a perfect flagship species for Cebu, as it occurs in a wide variety of habitats, and the locals can actually recognize this bird. Thus, it is very iconic, and it is also territorial. Because it is a small, black, and skulking bird that loves the understory, it was also rather difficult to spot this species. It also has a patchy distribution and has limited population data. So what we lack in the ability to detect the species in a dark understory is compensated by its species reacting to the presence of people in the area. So it immediately makes a call or sings what we call give a melodious call, and then people can recognize and record this species. So we use again the synchronized counts, where we have the observers that are spaced at least 250 meters apart. We also do the simultaneous count starting at 6 a.m. and we combined the field data that we have generated with the historical records that we have of the sites to actually come up with an island-wide population density estimates. And our results have identified new localities and new subpopulations of the black shama. This has sort of tantamount to extending the range of occurrence of this species. In so doing, we were also able to determine the varieties of habitats where it can be found, as well as its preferred forest habitats. We also generated the island-wide population density estimates, and this has provided the strong evidence that the population is actually increasing. But here's where it has actually become powerful. The conservation of the primary habitat and the species itself was led and sustained by a local community called Kapunungan sa mga mag-uumas sa Yutang Lasangnon sa Bululakaw, Nugas Alcoy, or KMYLBNA. The KMYLBNA is a people's organization organized by the DNR and was given a tenure of 25 years to manage 2,600 hectares of forest land. This is through an agreement called Community-Based Forest Management Agreement, CBFMA, through the DNR. Within these forest lands, the members of the KMYLBNA will have the right to manage and till the land, have their housing, and manage the assigned portion of this forest land. As long as this is within the guidelines of the approved management plan, these are the people can make use of this area for 25 years. And in 25 years of managing the forest land, what was once a badly degraded second growth area in 1985 has now been fully restored as a mature forest over limestone or limestone forest of more than 2,000 hectares. This is actually a satellite image that shows how this forest has changed over time. Take note, it's a satellite image. So you can see the power of these communities supporting the largest remaining forest and the largest population of the threatened endemic species of Cebu. Technically, it's biodiversity. And it is the smallest town of the island. Because of this combined evidence, the black shama was downlisted from endangered to least concern. And the Cebu bubok actually benefited from the protection of the black shama habitat and the black shama itself. And it also has improved its conservation status from endangered to vulnerable. So I would just like to emphasize that this field data combined with community action has led to a global conservation status change. This is what successful conservation translation looks like. Now let's move to a more difficult scenario. We are actually looking at a highly fragmented landscape of the southwestern key biodiversity area of Negros. This is home to the critically endangered, based on DNR DAO, yellow-faced flameback, an endangered species according to IUCN, and the Philippine tube-nosed fruit bat and the Visayan warty pig, among many other species that are also unique to Negros and in the southern portion of the islands. The challenge in doing monitoring and data collection for this site is that it is very remote. And the habitats that are found here are also very isolated. And we have very limited data that we can gather because of these challenges. And added to this, there are many ancestral domain claims and also ancestral domain titles that has been issued here. So there are other different conservation modalities that we have to attend to, and in considering that the rest of the forest land remains unprotected. So we relied heavily on the presence and absence recording of this species, and the local knowledge of the indigenous people and the local communities. This was not perfect data, but it was sufficient and aligned with management needs. We were then able to identify the critical habitat patches and map out the potential wildlife movement to design a functional connectivity strategy that would actually promote biodiversity, address impacts of climate change that is also a nature-based solution. And this strategy also links the forests of Balinsasayao Twin Lakes Natural Park and Cuernos de Negros mountain range to the smaller patches of Santa Catalina here, and also to the other patches of forest from Butuan all the way up to Cauayan. And this became the basis for an emerald necklace conservation strategy. Our key insight in here is that even imperfect data, if aligned with management needs, can actually inform policy decisions. We now shift our attention to the wetlands. I have to bring this up as wetlands play an important role in mitigating the impacts of flooding and sea level rise. Wetlands are extensive, dynamic, and complex ecosystems. And it is perfect for citizen science monitoring program. For over a decade, monitoring of the Negros Occidental coastal wetlands has generated the consistent data for the species of birds and waterbirds that are found in these sites. As this involves 3 cities and 7 municipalities, imagine counting all the waterbirds would require a lot of resources and a lot of volunteers. Enlisting the support of the local government from the provincial to the city and municipal level is very, very important. With DNR leading the count, PhilBio provided the training for citizen science. We also co-developed the monitoring design and co-led the implementation of the annual counts. The data that was built in for more than 10 years has significantly changed the conservation importance of the coastal wetlands of Negros. The highest number of individuals recorded has reached more than 50,000 in 2014. And these sites have the highest concentrations of endangered great nuts and near-threatened black-tailed godwits and other threatened and near-threatened waterbirds for more than 80 years. The local government has supported this waterbird count as this forms part of the regular monitoring under the wetland ecosystem required under the seal of good local governance. The LGUs and local communities has also realized the value of a healthy wetland and that it is tightly linked with their source of livelihood. Many of the high-value shellfishes actually come from the Nokwaka wetlands. So it's not just birds, it's also food security. So where does it all have led to? This has led to the designation of Negros Occidental Coastal Wetlands Conservation Area, we call this actually Nokwaka, as the 7th wetland of international importance, or a Ramsar site, and also the 4th East Asian Australasian Flyway Network site. Imagine that it's been quite a long time since the last Ramsar and last East Asian Flyway Network site was actually declared. And that drought was filled in by Nokwaka, and this was replicated, this methodology, this system, this strategy replicated in the Mindanao sites, and that it just also has led to new Ramsar sites and new East Asian Flyway Network sites. We are also very proud that we have been part of the development and generation of the data of these sites, and also in assistance with our DNR and local partners on the sites. Our key message here is that local bird counts can actually contribute directly to global conservation policy frameworks. So, sorry. From these experiences, 5 principles have emerged: simplicity, relevance, scalability, credibility, and accessibility. Methods must be easy to implement and repeat. Data must answer management questions. Involvement of communities and citizen science allows it to be scaled up to a landscape and seascape level. Scientifically sound and defensible, and shared with decision makers in usable formats. I am now on my last 2 slides, so let me end where we started. It's all about collecting the right data and ensuring it reaches the people who make the decisions. Remember that count data can define protection priorities, and that our field observations can actually reshape conservation strategies, and that community monitoring can influence global policy. So the question is no longer whether we can generate data. I think the question is, are we generating the right data and ensuring it reaches those who make decisions? Because this is how we make science count. Maraming salamat po. Thank you for listening.
Director Lisa, thank you for that profound and deeply grounded presentation. And now we will open the floor for questioning to both Dr. Jason Ibanez and Director Lisa. This is a wonderful opportunity for you to address your questions. Okay. We would like to request Dr. Ibanez, sir, for the question. All right. Who would like to ask question first? Anyone? How about question for Director Lisa? Wala po bang magtatanong? Okay.
Hello, good afternoon. I'm April again from Caraga State University. My question is directed to Dr. Jason Ibanez of the Philippine Eagle. Sir, you made mention earlier about the suitability count for the new method that you or that your other colleagues have done. Since the Philippine eagle only goes back to its habitat and its nest over and over again, how about the other species? Are there any other species that could benefit those type of method?
Yes, well, thank you for that question. I guess I think all of many of the raptor species can benefit from this type of survey method because many of them, including the southern Philippine hawk eagle, no, the Philippine serpent eagle, they're also very loyal to the places where they breed. So it's just a matter of identifying a new minimum survey size, area size. So for instance, for the Philippine eagle, we found out through telemetry that the average home range size is 7,300 hectares, but only a certain percentage of that, less than 50%, is used repeatedly or extensively used. So that was the reason why we only adopted yung 3,600 hectares as a minimum size for the hexagon. Now it would be a different case for other raptors. I believe that for the hawk eagle, the territory is smaller.
Um, and interestingly, we also found an active Philippine eagle nest within a Philippine eagle nesting site. So, and what's really nice about the survey method is that while we're only looking or while our focus is looking at Philippine eagles or finding whether there are Philippine eagles or not within the hexagon, because other raptors are also visible during the survey period, we're also listing down yung mga other raptors that are seen. So for instance, in the latest survey that we did in Cabuaya, Mati, they've detected the Philippine eagle plus 9 more species of raptors. So the beauty of this method is that you're also generating baselines for other diurnal raptors. And I'm pleased to share that the methodology has been adopted already by the Biodiversity Management Bureau as an official survey method for Philippine eagles and raptors. And it's already part of their compendium of endorsed methods for biodiversity surveys. I believe yung methods din ni na Lisa is already part of that compendium. So yes, anyone who might be interested to tweak the survey method for other raptors, you're very welcome to do it, and we'll be happy to assist.
Thank you so much for the very comprehensive answer, sir.
Good afternoon, Sir Jason. I'm from Father's Trinorius University po, from Morelos campus, and I would like to ask a question po about the Raptors po, sir. And how they, if someone, sir, would try to hunt them down, sir, and mapatay nila ang eagle nga gi-track, sir, how would we know nga ipatay sila sa hunter, and what would be the consequences sa kanang pagpatay, sir? Thank you, sir.
Yes, well, thank you for that very important question. No? So the telemetry study or putting GPS and radio transmitters is very important because it also allows you to determine yung status ng bird. So for instance, kapag nabaril yung bird with the radio transmitter, because of the lack of movement for the next 6 hours, the radio transmitter would switch to what they call the mortality mode. Okay, so for a moving bird, the signal coming from the radio transmitter is 60 bits per minute. Okay, so marireceive mo yung bits na yan through your receiver. And so kahit hindi mo makita yung bird, as long as 60 bits per minute siya, that means it's alive. But if it's dead or it's not moving, or pwede ding yung transmitter mo fell down from the bird, it would trigger what they call the mortality mode, where madodoble yung speed ng beat to 120 beats per minute. So kapag nagmo-monitor kami, yun talaga yung ayaw naming marinig, yung mortality mode. And many of our cases, may mga cases din, mostly female eagles, because female eagles are larger and more aggressive and more dominant, woman power, Philippine eagles, they can actually tear off yung kanilang harness. So may cases kami na nahulog yung transmitter. And then mortality mode yung reading, but we were relieved to know na naputol lang pala. Because that's a better outcome, no? Yung mahulog lang yung transmitter kaysa makita mo yung decomposing bird or rotting bird. So that's very useful. No? Yun I think beauty nga ng radio transmitter. It can tell you the state of the bird. In terms of penalties, well, in fact, medyo stiff yung penalty, no? Strict yung penalty ng Wildlife Resources Conservation Protection Act. If you shot and killed a Philippine eagle, which is a critically endangered species, or any critically endangered species, you'll be jailed for 12 years and penalized on top of yung imprisonment, 2 million pesos. Okay, so actually yung law natin satisfies yung severity test. Very severe ang punishment. But I think the real issue is enforcement. Doon talaga tayo nag-fail enforcement. And again, if I may share, part of the reforms that we really need to do, if we really want the law to be effective, is to really do enforcement seriously. And that's why for the Philippine Eagle Conservation Program, a very important role that we're looking at would be the role of the Philippine National Police to enforce yung firearms law. It can really make a difference in terms of yung shooting and killing ng not only Philippine Eagles but wildlife. But equally important also is working with the communities where the Philippine Eagle is nesting. So two-way siya. You invest on community education, incentivizing conservation. They see they're better off conserving than shooting or killing the bird. But also at the same time, sabayan ng strict law enforcement.
I was also wondering po, sir, po, about ito sa Philippine eagle nga it's still considered as an endangered species po. Unsa di ang rate is sa pag what would be the year ng balik na ito sa normal ang ilang population?
Right. Well, that's really a very nice question. And sadly, to be honest, we couldn't really downgrade yung status ng Philippine Eagle from critically endangered to endangered within our lifetime. Kasi by design and by evolution and by yung natural population size niya, Philippine Eagles have historically been as small, they already had small population kasi they're apex predators. Now, considering the Philippines as a small archipelago, based on our calculations, the most number of eagles we had pre-colonial is more or less 2,000 pairs. So, kumbaga, konti na sila daan. And unfortunately, we only have 400 pairs left, less than 400 pairs. So that's 80% reduction in the original population size. If we assume that in the past, we have 2,000 adult pairs. Now, for the endangered status, kailangan ang total number mo would be 50% of the original, which means 1,000 pairs. Okay, so 400 pairs versus 1,000 pairs. And they're forest dependent. So if we really want to downgrade yung status niya from critically endangered to endangered, we need to more than double yung population number. And also, more than double yung amount of forest habitat that we have. But all hope is not lost. What we can practically adopt is to improve the conditions of every nesting territory available within the Philippines. If you have 400 pairs, a maybe doable target would be 45% of this population, especially yung mga population, yung mga territories that are threatened, ma-reduce mo yung threat. 45%. So that's yung parang minimum. So that gives you kumbaga, a guiding star on how to practically conserve the eagle. No, so yun nga, it doesn't mean we cannot help the species, but we can improve yung conditions niya. And maybe if we do this consistently, 2 generations or 3 generations from now, we can make a difference. We can downgrade yung status niya from critically endangered to endangered, and also helping all other species found within the territory of the Philippines.
One last question, sir. Was it also because po sir nga kuan nga mga nang gamay ilang population, sir, because of hunting, sir, or other factors?
Yes, primarily hunting and shooting. Yun talaga ang challenge mo for an apex predator. Again, we can lose eagles even though we have suitable habitat. Habitat loss is the second most important factor. Disease can be another factor. Bird flu, and then yung Newcastle's disease. Kaya namin nilipat yung mga breeding birds namin from the old facility to an isolated facility because of yung threat ng bird flu. And then, of course, there's climate change. Pero all of these threats are also facing humanity. I think yung Philippine eagle is a barometer of what the human species is also facing. So if we tackle these issues, in the long run, we're also addressing threats that are also threatening our very own existence. And that's why the Philippine eagle is a very nice flagship and symbol for healing nature, because it can tell us whether we're doing it right or we're doing it wrong.
Thank you, sir. Madiyong hapon, good afternoon po. Alvin from National Museum of the Philippines, Butuan. I would like to direct my question to both plenary speakers po, as I noted the involvement of local communities in your presentation. So could you please elaborate on how this engagement was carried out and how indigenous knowledge was integrated into the conservation effort and the overall process po nung ginagawa niyo po? Thank you.
Lisa, would you like to answer first?
Presentation.
Thank
I
you.
really enjoyed it.
Likewise.
I was also listening
Likewise.
to it. Local communities. Local communities with PhilBio is always an integral part ng kanyang work. In fact, we never do things without the participation of at least four na sectors. So these are the local communities and indigenous people, the local government, mandated agencies, and the academe private sector and the like. Mostly it's more of academe. Now we have a few of the private sector. So within this circle, the local communities is very important since they are on the ground, they are on the front line, and they are your first line of defense. When the communities are actually benefiting directly from their habitat, or they are directly benefiting, I'm gonna move because I think hinay ang ako internet. If they are directly benefiting from their uh, what's this from the resources at hand, then they would actually be more supportive of the conservation program. And if the communities also sees and builds with that pride of what they have, they will also be likely the first person to actually support it. So when you have the communities supporting the conservation of your species and the habitat, it can also translate to political votes. So if the people demands for the protection of their habitat and for protection of their species, then they can actually influence policy and influence the decision of that area. So is the indigenous people. So right from the very beginning, what the local communities thinks that and also practices that is sustainable, or they feel that should be right for their ecosystem, should also be part of the main basis of doing the conservation program. After all, what is our parang ano ba yung karapatan natin magsabi ko ano yung pwede at hindi? Hindi naman tayo ang nakatira doon. Many cases, local communities are also very supportive once they understand. So understanding the value of the ecosystems, the value of the species, and what's in it for them will also help move the direction of the conservation program. So I think Jason can expand more on this. Thank you for the question, by the way.
Yes. Well, thank you, Lisa. Again, I would like to echo also what Ma'am Lisa mentioned. No, it's the same in the case of the Philippine Eagle conservation case. No, as she pointed out, there should be an alignment of yung aspirations ng IPs with the conservation that you're trying to do. Secondly, they should be part of it. Okay, they should be part of the decision making. And then thirdly, they should benefit, including material benefits, no, financial benefits from conservation. So, siguro just to give some detail, for the Philippine Eagle conservation case, we make sure that we understand yung indigenous plan, because the ADS-DPP or the indigenous plan, you will find yung aspirations nila for the ancestral domain. And in many of the ADS-DPPs we've reviewed, forest conservation is part and parcel of the target. No? But of course, there's a layer of cultural values for the same land. So, and remarkably, the same nesting territory, the same nesting site, is also the same sacred forest, the same watershed, the same burial ground, the same taboo area that they want to protect. So that's where you can find yung alignment. Secondly, is rights-based. No, they have the right to decide on how to use their land. So it's very important to situate yung conservation mo with the decision-making powers of the indigenous political structure, yung mga elders, because they're the one making the decisions. No, if in the mainstream you have yung legislative, executive, and judiciary, sa mga indigenous peoples, yung IPS mo holds all of those functions. No, they do decision making on land use. They impose yung mga penalties if you violate the rules based on the traditional justice system na husay. And then again, I think this is very important, is to make sure that if they give service and they share their knowledge, we pay them fairly for the services and knowledge that they provide. We should consider it as a legitimate knowledge and skill in the same way that we pay for people that we hire and for consultants that we engage when they share their knowledge. And that's a very important element of conservation kasi that can now incentivize yung perpetuation ng culture. And so that's how practically we can empower and support indigenous communities in conservation.
Good afternoon po, Glenn from the Fed. Pero, satisfied na po ba tayo, sir, sa mga data na nag-gather natin?
Yes. Well, there's no harm in asking for more updated data. But I think what's really important is for us to make the most out of the best data that we have. I think that would really matter for endangered species. Again, in the real world, kulang tayo ng funding, kulang tayo ng resources, manpower, minsan hindi siya priority ng government. Pero we still work with what we have. I really like this conference sponsored and co-convened by our colleagues from FSUU. Because what it's trying to do or will do is to make data accessible to everyone. And just imagine how empowering that is. No? If the species distribution models or maps can be used by the local government so that it would inform yung kanilang land use planning. Or if yung mga species data on where wildlife species, endangered species would occur, that can be used by others for conservation, including the DNR. So I think yun yung beauty yun. Again, if we have the resources, the more the merrier. Pero again, in the real world, we really need to work with what we have
and make.
How about po sa technology natin? Meron ba kayong nais makamit or i-acquire para mas ma-monitor pa natin at maproteksyonan ang mga species na ito? Aside from the GPS,
Yes. Maybe Lisa, ano bang mga wish list mo dyan for mga technology or tools?
Or coming from mandated agencies. It's yung mismong pagka-involve mo doon sa mga activities is very empowering. It connects you directly to the species, to the ecosystem, to the people. And pag ganun na level yung pag-provide ng experience doon sa mga taong dapat kasama doon sa management, that really brings the level ng playing field at a higher standard. Kasi pag malapit na sa puso, may taya na doon, mas marami ng mas hindi na niya makakalimutan. So, yan. Pag-provide ng experience, so that means kailangan dalhin natin doon yung mga tao sa field or at least provide an opportunity whenever it's possible. If not, I like the idea of what the Philippine Eagle has started and some other groups na may remote nila na camera na nakamonitor doon sa species. Kasi that gives the public the access to the species, no, what's going on, and to be involved, to be part of that whole thing. Maganda din sigurong mabigyan ng opportunity na tingnan kung paano natin gamitin yung mga new technologies ngayon kung saan mas mapabilis yung pag-analyze ng data. Alam kong marami dyan na nakaupo din at nakikinig sa interaction natin. We have lots and loads of data and they are either sitting in papers, in transect forms, in reports, and in the shelves or in the notebooks na hindi pa napaprocess. And having that technology or that ability to actually process those information and then directly links and feed that back to science, that would be wonderful. If we can have that kind of technology and also access to that technology, kasi meron mahal lang at hindi lang siya ganun ka-accessible.
Hello, Lisa. Hindi ako mag-question. Just to respond dun siguro sa sa question. Yeah, it's right. Technology is quite expensive. That's why before we use those, we need to be clear about our objective. Hindi dahil may technology ka, you can do everything. Masasayang yung resources mo if you do not think over your objectives. You have to be clear of where you are going to use that kind of technology. You need to look at ano yung questions na gusto mong isagot and what is the appropriate technology to answer that question. I think that's technology pag may pera ka, madali na lang yan. But answering the right question or asking the right question, answering the right question, using that right technology is important before we move to that kind of approach. Yun lang po.
Good afternoon, everyone. Good afternoon, Mom Lisa. You've mentioned earlier, Ma'am, that sometimes there are times na kapag nagagather kayo ng data, it's limited and you can just gather data as much as you can kung kayang mag-gather ng data. So, given the situation, Ma'am, how can we ensure that the data gathered are useful or like somewhat reliable or credible given that situation, Ma'am, na limited sometimes ang pag-collect natin ng data. By the way, I'm Isopro from Father Saturnino Orius Morelos po. Thank
Sort of questions you are actually going to answer. And in our case, kasi hindi lang siya about sa research, it's also about sa conservation. We have a medium-term and long-term plan about how we want to see or what kind of progress do we want to see sa conservation ng sites, ng species, or yung management ng isang lugar. So, with that in mind, alam mo, ang buhay ng isang NGO medyo challenging because we don't have that yung privilege na gagawa ka lang ng research and then hanggang doon, or at least focus ka lang sa research. For us, we have to embed the research and do that as part of your conservation plan. Having said that, we now have that medium term at long term na pagtingin kung ano yung gagawin sa lugar. So we take in the projects, the request, at yung mga gagawin na mga monitoring na mga short term doon sa mga places na nandoon din yung species and ecosystems or place of interest for the conservation of Philippines biodiversity according to the priorities ng PhilBio. In that way, we don't waste so much resources and time by just picking up all of those information from the field. Ang naging challenge namin ay kasi marami yung gagawin na yan, pakonti-konti, tatagal din medyo matagal yung panahon na ma-build mo yung information. And that is why we have to do an extra work of doing the partnerships, involving the citizen sciences, and also the local government and mandated agencies para naka-align doon sa regular program. So when it is done that way, I think kahit na contribution siya ng ibang mga group, as long as they follow the same methodology, they follow the same levels of standards, the data can actually be accumulated and comparable. I hope I answered the question.
Good afternoon, everyone. Good afternoon po, Sir Jason and Ma'am Lisa. I'm actually fanboying right now because your names, both of your names, are really that I consider as icons in terms of conservation and biodiversity in the Philippines. Our school, actually, Philippine Science High School in Caraga, is actively participating in wildlife quiz bowls in the past 3 years here in Caraga region. I would like to ask some questions first to Sir Jason and then later for Ma'am Lisa. Sir Jason, you mentioned earlier, sir, that one of the problems we have right now in terms of conservation of the Philippine eagle, to be more specific, is its Philippine eagles, you found out it has low genetic diversity. Diba, sir? So, and then I would like to ask if are there any steps or efforts that we are actually doing right now to improve this genetic diversity or preserve this genetic diversity of the Philippine eagle, and how can technology aid in this effort?
Yes, well, that's a very nice and strategic question, ano? And yes, I'm excited to share that, again, based on the two current genomic mapping research, it turns out that conservation breeding is really effective at restoring genetic diversity. And what's really nice about the conservation breeding program is that prior to the genomic mapping study, the decision of our pioneers and colleagues was based on the simple idea that you don't pair eagles coming from the same mountain range. And again, conservation breeding started as early as the 80s with Pag-asa as the first captive bred bird. And they followed that protocol. Pag may bagong bird na pumapasok, you look at yung kanyang origins, and then you don't pair him or her to an eagle that's from the same place. And that simple decision showed that, as shown in my graph, there's nearly zero inbreeding coefficient for all eagles that were bred in captivity. And in addition to that, yung captive offsprings mo represent 17 haplotypes, 4 of which are absent from the birds that were rescued from the wild. So it shows that, again, in terms of genetic diversity, conservation breeding is doing its role. And we were affirmed by the genomic study. Now that we have the genome maps of all the birds, and based on the wisdom of the study, in order for us to create a pool, a gene pool, where all, if possible, all bloodlines are represented as an insurance policy in captivity, and interbreed itong mga birds na to so that you produce more combinations, and that again contributes to genetic diversity. We really need to get at least one representative from every potential bloodline in the wild. And we're very happy to share that it's now part and parcel of the Species Action Plan. And the next steps for the Philippine Eagle Foundation is to get, together with our DNR colleagues, to get representation from the wild, rather than just let them get shot or trapped in the wild. Find, secure a genetic representative, breed them in captivity, with conservation breeding as one strategy to maintain genetic diversity while we improve habitat conditions and wild population conditions in the natural setting. So we're now moving towards that, and I'm pleased to share that institutions like Manday Nature in Singapore and also Czech Republic and European partners are now on board and would help fund and also support itong initiative. And thanks for raising that, because again, that's a response to a study that came out, and the national government is also supporting that action as a result of the baseline data generation.
Thank you so much, sir, for your answer. To Ma'am Lisa, hi ma'am, good afternoon po. Ma'am, you are very experienced na po in the field. You have so many years na doing field works. Ma'am, in your many years in the field, are there any case or any cases when a program, whether government or private program, which could have been ecologically detrimental, that has been challenged or stopped due to the results of your field work? And we would like to hear some anecdotes or some story from you.
Up because of the results of our fieldwork. Okay, paano ko yan i-context? So, I have an experience before when we were doing the conservation work, helping the communities, assisting the communities of Alcoy for the protection of that black shama habitat or the Cebu flower pecker habitat. And it so happened that the local government at the time wanted to do road development right next to a portion of that forest patch will actually be affected. And because we have mapped out the geotagged all the trees of the critically endangered Cebu cinnamon and also the territories of the black shama, we were able to overlay the distribution of the Cebu black shama territories and the Cebu cinnamon, and then present that as part of the results to argue why the road development should not be there. So while we did not contest the local government in terms of making a protest about this one, we were just simply laying down the consequences, the impact of this to the biodiversity of that area. And we were almost persona non grata with the local government at that time. It was also a good thing because we have been closely working with them, and that half of that group, or at least several key members of that council, actually supports the conservation program that we initiated or co-initiated with the local communities. So in the end, we struggled for about 6 months, but we were able to get past through that. In the end, the local communities held their ground, and they were the ones who told the local council that they will not be supporting this kind of work. So they ended up giving up that road development project and saving the forest. So hindi siya necessarily because of the results siguro ng project na na-stop yun. In fact, naging rason pa siya kung bakit na-stop yung kanilang pinopropose na program. There were also cases na may mga population estimates kaming ginagawa sa mga threatened species, and sometimes people do not parang have a hard time accepting na ito yung number. Sometimes kasi mas malaki yung number na nabibilang at hindi yun ang ine-expect nila na number. So I think it led to some misunderstanding, or at least parang I think acceptance. It needs to have time for people to accept. Maybe another group to also help validate that population estimates and data. And so that more people can actually provide the information and challenge what is actually currently yung pinopropose na population status. People sometimes think na pag nag-change yung conservation status ng species, mawawala na yung attention doon sa species na yun. Yun ang masaklap kay science. Wala kasi siyang feelings. Based lang siya doon sa numbers. So we have to trust the data, and until such time that new data will come in and prove it otherwise, we will stand by the data. There was also an instant. So this was the first time we did the survey as a graduate student in Alcoy. No, not in Alcoy, in the northern part of Cebu. And, 20 years, sinusundan ako ni Dr. Angel Alcala. And every time we see, at the time, parang alam ko na kung anong itatanong sa akin ni Doc Aka. Tanungin niya sa akin kung totoo ba talagang nahuli namin ang Philippine bareback fruit bat. Because he does not believe that we actually caught the Philippine bareback fruit bat because we did not collect the specimen. So this was way back in year 2001, and we were the first team. We were just a group of students who were just out there doing some field work in the northern part of Cebu, a badly degraded forest. Nobody expected anything. And then we caught 3 individuals of the Philippine bareback fruit bat, or the Visayan bareback fruit bat. At the time, it was extinct. Nobody has seen this species, at least with the group who am I with. So, yung nakuha namin yun, nahuli namin yun, part of the team do not want to release the species or the individuals because they suspect na wala tayong ebidensya, mga estudyante lang tayo. At kailangan nating magkaroon ng ebidensya para sure tayo na na-rediscover natin. My position at the time was that, no, release it back. We are doing this primarily for the conservation of species and the conservation of biodiversity ng Cebu or Philippines. That's the reason why we are doing the survey in the first place. And why do we need to collect that individual? And we will be the reason for its final extinction. So we went into a lot of arguments, and in the end, there was a lot of mixed reactions at the time because we took a lot of photos. Well, we did not have that enough specimen. Nobody have thought about getting a hair sample or something just for DNA purposes in the future. And but then we just took some photos and then released the animal. So it took a while before all of this. A lot of things still went through, even to the point of publication, because we waited for at least 3 to 4 years before we actually published. And there was a reason for that. The moment that we had the news that the bareback fruit bat was rediscovered, we talked to the local, our partner was DNR, and we talked to DNR at the time to be subtle about this and not ensure first the protection on the caves and the habitats in this place before we announce. And but there was an announcement, there was a press release, and people were so excited about this. We were half happy about that, and we understand their excitement. But what happened after was I was informed by a friend of mine from Germany that there was a pet magazine that he picked up on the train in Germany that has a notification there that they're selling, or somebody who is interested to get hold of this critically endangered, newly rediscovered bareback fruit bat, dead or alive. So immediately that alerted us. So what we did was we went back to the sites a few months after we rediscovered this. We worked with the local government there, with the local communities, and started all this conservation program for the cave conservation. We took note of some of the incidents of increased hunting activities and people that are searching for bats, and then a lot of bats were actually caught and died in that process because they were not they were just trying to get any bat and then select from whatever that was captured. So it was not good. But in the end, when we laid down the conservation on the site, when we were ensured that the protection of the caves were there at that time, that's time when we started publishing the data. And even if there were a lot of questions about that data, for us, it's that we stand by with what we have and what we know. And for the information, it would be up to the next one to refute that. Thank you.
Thank you very much, ma'am, for the comprehensive answer. Thank you so much po, sir. Mabuhay po kayo.
Sir, there is a All right. So we would like to thank you for the incisive, thoughtful, and passionate questions. The dialogue between field scientists and the community is exactly the spirit of this Congress was designed to foster. And now may we call on Dr. Shirley and Dr. Shirley to give the certificate to Dr. Ibanez and Director Lisa. But allow me first to read the content of the certificate. Father Saturnino Orius University, Butuan City, Philippines, Certificate of Appreciation is awarded to Dr. Jason C. Ibanez for delivering an engaging talk at the plenary session on field-based biodiversity conservation during the first Philippine Biodiversity Data Congress, with the theme Field to Cloud, the future of open access conservation. Given this 8th day of April 2026, TB Function Hall, Father Saturnino Orius University, signed by University President, Reverend Father Randy Jasper COJ. And also, Certificate of Appreciation is awarded to Director Lisa J. Paguntalan Marte for delivering engaging talk at the plenary session of field-based biodiversity conservation during the first Philippine Biodiversity Data Congress with the theme Field of Cloud, the future to open access conservation, given this 8th day of April 2026. Signed by the University President, Reverend Father Randy Chester COJ.
Maraming salamat po.
As we approach to the close of our plenary session, it is now time to gather the threads of everything we have heard today and weave them into a coherent picture of where biodiversity science in the Philippines stands and where it must go. To give the synthesis, we would like to call the coordinator of Biodiversity and Informatics Research Center, or BIRC, of Father Saturnino Orius University, whose contribution to biodiversity research in Mindanao and across the Philippines are extensive and highly regarded. Under her leadership, BIRC has become a hub of scientific excellence and a vital contributor of our national knowledge of biodiversity. Please join me in welcoming the coordinator of FSUU BIRC, Dr. Marites Bonachita Sangela. Ma'am.
Thank you, Sir Romel. Good afternoon, everyone. Mata pa. Okay, so for the first installation of the Philippine Biodiversity Congress, with the theme From Field to Cloud, the Future of Open Access Conservation, we are indeed thankful for the opportunity to connect with scientists and conservationists and with each other to learn the significance of gathering correct data and informing the public about these data effectively. Dr. Rafe Brown of the University of Kansas, our first speaker, informed us of the correct and ethical methodologies to note the most complete natural history data sets available for us in the field and how to effectively store them via open access digital platforms with data aggregators such as the Global Biodiversity Information Facility and the IDIG Bio. This is to ensure integrity, transparency, and repeatability for data consumers. As such, data is freely available and analysis is comprehensive, allowing us to produce a more complete picture of the changes in life forms on our planet. Our second speaker, Dr. Duncan Ershik from the University of Massachusetts Amherst, discussed with us contemporary tools for recording biological information of the natural world, intersecting the fields of art, biology, and computer science. With this, the creation of open access 3D models can be used by stakeholders worldwide. Connected digitally, we can now use these 3D models as tools to describe the natural history of an organism right at our fingertips, to describe an organism's body size and length, mass, volume in relation to its biological concepts such as functional morphology, behavior, and ecology. In the field of ecology, our third speaker, Dr. Aloy Duya, has shown us that the implementation of technology-driven tools to digitally record an organism's natural habitat, movement, or population counts must be complemented with traditionally gathered data sets of the same to come up with comprehensive information for effective policy recommendations. In the field of conservation science, the implementation of effective species monitoring programs require not only dedication, but most importantly, rigorous multidisciplinary data and innovation. Our fourth speaker, Dr. Jason Ibanez, provided us with information gathered from the field, analyzed in the realm of biodiversity informatics, to model species distribution within predicted suitable habitats. This type of analysis allows efficient use of resources to support biodiversity programs, consequently reassuring effective implementation of conservation and the protection of the Philippine eagle and especially its forest habitats. On the other hand, Ms. Lisa Pagontalan discussed with us how we can use long-term data sets translated from the field to real change local and global conservation frameworks.
She emphasized that data gathered should significantly involve people, and this must be transformed into practical knowledge for the people. Overall, we had the privilege to gain more knowledge on our local biodiversity, which sparked productive discussions on biodiversity documentation and digitization. Indeed, the first Biodiversity Data Congress is about the future of biodiversity conservation in the world of open access and digital information. Thank you and have a great rest of your day.
Thank you, Dr. Sagela. And now, time has come to deliver the closing remarks. We are honored to welcome the Director for Graduate Studies and Research of Father Saturnino Orius University, a leader whose dedication to building a culture of research at FSUU. Please help me welcome Dr. Shirlene Midore T. Alegre. Ma'am.
Good afternoon to everyone participating on site and via our FSUU FB page live stream. On behalf of Father Saturnino Urios University, I would like to extend our sincere gratitude to the different committees who made the Philippine Biodiversity Data Congress 2026 possible. To the organizing committee, headed by the Biodiversity Informatics and Research Center Coordinator, Dr. Marites Sanguila, in coordination with our Vice President for Academic Affairs and Research, Dr. Arlene Floreta, thank you for your leadership and careful planning. To Sir Dominic Giritan and the Project Mabulig team, we deeply appreciate your expertise in upholding the quality and integrity of the research presented. To the logistics team, Sir Jeffrey Carin, Sir Ivan Chris de Labahan, Miss Kimberly Derama, Dr. Mary Grace Medrano, Sir Raniel Ronquillo, and Miss CJ de la Peña. The plenary and program committee members, Ma'am Lauren Grace Haranilla, Ma'am Jessan V. Cordova, Sir Romel Coquilla, and Miss Antoinette de la Peña. The articulation and technical team, and Sir June Castillo of ITDM, and the Strategic Communications Office, headed by Ma'am Suzette Viñas. Your dedication ensured the smooth flow of every session. To the in charge of the booths and workshops, Mr. Alvin Amarille, Miss Daphne Kale Bahoy, Miss Lee Angela Garnica, Miss Ana Cristina Labaja, Mr. J. Lauzon, Miss Daphne Jane Nato, Mr. Brevin Peter Tagsa, Mr. Fritz Villahermosa, the BIRC Undergraduate Research Experience, BS Entertainment and Multimedia Computing, Computer Studies Program headed by the Dean, Mr. Lambert Buligor, and the Young Biologists and Mathematics Society, and to all staff and volunteers, your invaluable support made this event truly successful. We also extend our heartfelt thanks to our distinguished resource speakers, Dr. Rafe Brown of Kansas University Biodiversity Institute, Dr. Duncan Ershek of Digital Life of the University of Massachusetts Amherst, Dr. Mariano Roy Duya of UP Diliman Institute of Biology, who is here with us today. Thank you very much, Dr. Duya. And Dr. Jason Ibanez of the Philippine Eagle Foundation, and Director Lisa Paguntalan Marte of the Philippine Biodiversity Conservation Foundation, Incorporated, for generously sharing your knowledge and insights about biodiversity. The DOSTP shared through ETDD Chief Ma'am Edna Nascianceno, and to the FSUU management team, led by our University President, Reverend Father Randy Jasper C. Ochige. Thank you for your guidance and unwavering support. To the students, faculty, and participants from the different colleges and universities across the region and all over the Philippines and abroad, we deeply appreciated your enthusiasm, curiosity, and commitment to advancing research. As we celebrate this Congress, we are reminded of the vital role research plays in addressing today's global challenges. The preservation of biodiversity remains essential to sustaining life and ecosystems, while digitization continues to transform how we generate, share, and utilize knowledge. Equally important is the commitment to open access, which ensures that information is inclusive, accessible, and beneficial to a wider community. This gathering, the PBDC 2026, reflects not only academic excellence, but also a shared responsibility for all of us to advance knowledge for the greater good. Thank you for promoting a space where these important ideas can thrive. Once again, our sincere gratitude to all of you. Have a great day ahead and see you in PBDC 2028. Thank you once again.
Dr. Alegre, thank you for those heartfelt and visionary closing words. And now, ladies and gentlemen, this marks the conclusion of our plenary program and end of today's live stream broadcast. This is Rommel has been said, mayong hapon sa inyong tanan. By the way, before you exit, we would require you to scan the QR for you to receive the certificate. Okay, please answer or fill in the survey provided using the QR. You're just going to scan it in your using your mobile phone. Thank you. By the way, your certificate will automatically send to your email. The email that you are using. University, Agusan del Sur State University. We also have the Ateneo de Davao University. We have the Philippine Science High School, Department of Environment and Natural Resource Conservation and Development Division, National Museum of the Philippines, andito po sila, and also the Caraga State University Project Base. You may now proceed to second floor of CBE boardroom. CBE boardroom. Yung mga participant po natin para sa 3D modeling